Excellent. All right, I think we're going to get started. So good afternoon, everyone. On behalf of SaskSoil, I'd like to welcome everyone to the 33rd Annual Soil Health Conference, Why Carbon Matters. My name is Mark Heimer. I am the president of the SaskSoil Board of Directors. I'd like to begin by acknowledging that the land that we gather on today is Treaty 6 territory, a traditional territory of the Cree people and home of the Métis Nation. Whether you're joining us in person today or virtually, we would like to thank you for attending and supporting our conference. We've got a great lineup of speakers over the next day and a half, and we're very excited to see what they have to share and to see what kind of conversations get started because of it. As everyone knows, COVID-19 restrictions in Saskatchewan have been lifted, except for the mask mandate, which is still in place. I think we've been living with it long enough that everyone knows when you use your mask and when you don't. So if you could please comply with the rules that we've got so used to. We don't have much longer, but they're still in effect for now. Continuing education credits are available for this event. Exactly. There is a sign-up sheet at the registration table, and I'm told there will also be a QR code that will show up at some point throughout the day that you can use to scan as well. On the back of your name tags, there is a ticket that gives you the ability to claim a water infiltration kit. We are just finishing putting those together. They're not available right now, but keep checking back, and anyone who's interested in, we'll get one for you. All right. I would like to take this opportunity to thank our sponsors. Putting on a conference like this, it just wouldn't be possible without the support of businesses and organizations that believe in the messages and the types of things that we're trying to share with you. So their support is just, it's the only way things like this happen. So I would like to thank A&W, the Water Security Agency, Myers Norris Penny, General Mills, Fertos International Organic Incorporated, Alice New Acre Project, Western Grains Research Foundation, Vivagro Sustainable Solutions Incorporated, Union Forage, Sask Wheat, Performance Seed, Imperial Seed, Shelburne Reynolds, Sask Pulse, Sask Canola, Sask Oats, Sask Mustard, Saskatchewan Cattlemen Association, Grain Millers, Farm Credit Canada, Ducks Unlimited, Cows in Control, Angelic Land, Alfalfa Green, Aberhart's Ag Solutions, Zach's Home Hardware, and Golden West. And if I could get a round of applause for the sponsors, I think that would be fantastic. All right, at this time, I would like to ask SSCA board member Cody Straza to come up and introduce our speaker.
Hi, everyone. I'm Cody Straza, the Vice President of the Board of Directors at SaskSoil, and it's my pleasure to welcome our first speaker of the conference. Joel Williams is an independent plant and soil health educator and consultant who has worked in Australia, the UK, and Canada, where he is currently based. He has a keen interest in plant and soil interactions and how those may support the development of low-input farming solutions that maintain production and landscape function. Joel has a passion for teaching and sharing a combination of academic and practical information and has lectured to farming audiences internationally. Please help me welcome Joel Williams.
Thank you very much, Cody, and everybody at the association for helping out. I know we all know it's a lot of work to put together conferences, and there's a lot of work that goes on behind the scenes. So thank you very much to everybody. Okay, this is the first time that I've done this in 2 years. I feel almost a little bit nervous, funnily enough. First time I've worn trousers in 2 years, giving a talk as well. Normally doing a webinar, I just put on a shirt and can be wearing your PJ bottoms, you know, like, so there you go, dressed today. Yeah, what a nice feeling to be here and see faces, and hopefully we can have some time for discussion and questions a bit more intimately, which will be nice compared to the usual webinar format. So, and of course, hello to everybody who is still at home, tuning in virtually as well. So good to have you all here as well. So today is 2 topics, as I'm sure you've all seen the agenda that we're going to focus on. I've made the first one is going to be on nitrogen, and this one is going to be, I'll forewarn you, fairly dense. It's going to be a bit technical, a bit sciencey. We are going to go pretty deep into this topic. So we'll do this one first while I've got hopefully more of your attention. And the session this afternoon will be a little lighter. And there we're going to talk about how we can leverage the fertility in the soil. Of course, with fertilizer prices the way they are, there are nutrient reserves in your soil. So we'll talk about some of the biological strategies that we can use to unlock those, to leverage those, to optimize those, and make use of what's already there. So that'll be the kind of the core focus of the second session after break. So, and with that, let's dive in. And I'll have to keep my eye on the time. I've always struggled to prepare slides. Have I prepared too many? Have I prepared not enough? Trying to stick to the time. We'll see how we go. So it should be, let's see if I get this working. Okay. So the outline for this first session, we're really going to try and answer this question. Does nitrogen burn or build soil organic matter? I'll forewarn you now, it's not a straightforward answer. It's not a clear-cut answer there, but we're going to try and answer that question. Now, in order to answer that question, we really need to dive a little bit into soil organic matter, into soil carbon. So we're going to really lay the foundation of that first, and then we will move into answering the question. So we'll start off talking about roots, and roots really are the key to building soil organic matter. So we'll talk in length about roots. And then really the role of microbes, microbial biomass in processing those roots and turning them into soil organic matter and stabilizing the roots into soil carbon. So we're going to talk about the microbes. And then we'll bring into that a bit of discussions around root exudates, one of my favorite topics. We could talk about that for a long time. So we'll just bring in the context of root exudates there. And then we're going to start getting into this nitty-gritty. So I'm going to spend quite a bit of time on this point here, the types of organic matter or the quality of different soil organic matter. We do soil organic matter a great disservice when we talk about it as if it's one thing. Soil organic matter is many different things. There are many different types, many different qualities. It's not all uniform, and therefore the way in which those different types form, the way in which they're stabilized, we really should break them apart and talk about them in a very context-specific way. So that's where it's going to get into some nitty-gritty. And that will help us answer the question on nitrogen. And really from there, we'll move into talking about soil organic nitrogen. This is a really big part of this discussion as well. Something that is often overlooked. We don't seem to talk about this quite enough, I think. So we're going to dive deep into soil organic nitrogen, the organic forms of nitrogen in the soil. And those are very important to answer the question about nitrogen burning or building. So from there, we'll talk about the plant, move up to plants. We'll talk about the role of also organic nitrogen forms in plant nutrition. This is also a very emerging and interesting topic at the moment. There's a lot of rethinking happening and looking at these forms of organic nitrogen like amino acids and proteins and their contribution to plant growth. So we'll talk about that. And then round it off with some discussions on legumes, of course. Can't talk about all of this without talking about legumes, and a bit on legume companions. and then just broaden the discussion a little bit. What other effects does nitrogen have on, particularly on soil biota there? I'm going to have a little discussion there. And then very briefly round it off, which should open up to some questions, just a little bit of trying to get a little practical. So like I say, this is going to be pretty dense, and we'll have maybe the practical element through your questions. And I'll kind of just open the floor to that with just some practical strategies on how we can integrate nitrogen management strategies, integrate various strategies together to manage nitrogen. Okay, so that's going to be the flow. And let's see, 1:15 already, so let's go. Okay, so if we look historically at yields, and just two examples here, one from Europe and one from over here in the US, North America here, just this general obvious trend that yields have gone up steadily historically. Okay, they've been a bit plateauing maybe just recently, but if we look back, different time scales and whatnot, yields have been going up. Now, of course, we would therefore make the assumption that with those increased yields, there's been increased biomass production, therefore increased residue entering the soil. We should have seen, therefore, soil organic matter also increasing over the last 100-odd years of intensification of agriculture. But of course, we have not seen that generally, and it depends, context dependent here, but generally, soil organic matters have gone through quite a decline with the intensification of agriculture. Yet some nuances to that, of course, things like no-till and whatnot have helped, maybe a bit of nuance in that discussion, but there'd be a lot of agricultural soils that have declined in soil carbon or stayed the same, despite the fact that there's been a lot more residues, a lot of increasing yields, and more carbon coming into the system. And it's because it's not a simple equation. Just because there's more going in doesn't mean it's staying in. A lot of that carbon is ultimately oxidized off. As microbes break it down, they breathe, they respire, and they are releasing a lot of that carbon as CO2. Indeed, the vast majority of those crop residues do get released as CO2, as upwards of 75% of those residues that come in do ultimately just get breathed off. So we're left with only a much smaller fraction, 5, 10, 15%, that actually gets stabilized and stays in the soil. And that's what we're going to try and untangle to the rest of today's discussion. So one of the interesting things that we then start to think about here is that there's also a difference between perennials and annuals. We've also seen, of course, a shift to a lot of annual production. And the thing about annuals is they don't produce enough, they don't produce as much roots as perennials. And they don't produce as much root exudates. Annuals allocate a lot less carbon below ground. They keep more up and less down for the roots. Whereas perennials, they know they'll be here next year, so they're investing in root growth. They're investing for next year, so they will put more biomass down below into the roots. So here you can see also the green is perennials, a grassland example. We can see a big kind of peak of below ground allocation, rhizo deposits, root exudates. And we see that the green, the perennials, they have a lot more sustained kind of release over time. Whereas annuals, of course, they have a shorter life cycle. Of course, they have a very quick peak with the pink, you can see, and then a kind of general decline in their root activity, root exudates, et cetera, et cetera. Okay, so there's one factor there. The shift to annuals has meant less carbon going into the system. And there's just a nice visual to illustrate that. You can see on one side, we have perennials, we have nice big deep root systems, and annuals, generally we have smaller root systems. So there's a big factor that has led to these changes in soil organic carbon, this shift away from perenniality. And then we also have another factor, which is breeding. As we have bred for high yielding varieties with modern, with our breeding that arrived around the 50s and 60s and 70s with the Green Revolution there. What did that breeding do? It helped breed short varieties, which of course helped then ultimately drive biomass production, allocate biomass production upwards, above ground. Hence, we increase yields. Now, that's a trade-off. When we put more energy above ground biomass, we are putting less into below ground. So we can see this kind of, just in this example here, we're looking at Okay, one example, looking at comparing modern wheat varieties this paper is from Spain, but you can see here that right at this kind of transition of the Green Revolution, we started to increase above ground biomass, and we saw this little decline that came with a cost of less carbon allocation below ground to those roots. And we're kind of seeing this same kind of trend over here. So breeding, this was also another factor. We bred for high yield, we bred for above ground selection, above ground biomass, that's less roots. Nice visual also to this, just a simple little pot trial from this breeder from the UK. On the left, we have einkorn wheat, ancient variety, and on the right, we have modern durum. Okay, so same potting soil, planted on the same day, just a simple little visual experiment. You can see that clearly the einkorn wheat is allocating so much more carbon below ground. Okay, so much more roots on the left-hand side. And then we've also got this factor of fertilization. This has been another nuance, and that's what we're going to go much deeper into when we hit the nitrogen topic. We can see that what fertilizers generally have been very good at, what they have done is also to help drive above ground biomass. Fertilizers do help to increase yield, but that particularly is through a mechanism of allocating more carbon above ground. So here we have kind of lower or higher yielding systems here. As we increase fertilization intensity, you can see that yes, we get more above ground biomass, but actually the roots are kind of on par. Okay, we're not necessarily getting more root production just because we're getting more yield, is that kind of nuance as an effect of fertilization. And that's what we're going to dive deeper into in the nitrogen part of the discussion. So, you know, is soil organic matter really even that important? Here's a study here. I'll just read you a few points here, linking soil organic matter to yield. As it says here, although it is considered important to soil health, soil organic matter and the relationships with yield are a little bit more, there's a bit more gray area there. And so they did a big meta-analysis, a big study of looking at lots of other studies and trying to identify, find any links with, is soil organic matter even important as a factor towards yield? Generally, we associate it more strongly with soil health. And they found that on wheat and maize, on average, as we increase soil carbon, yes, there is a correlation with increasing yield up into about this 2%. Once we get above 2% soil carbon, that's where the relationship kind of plateaus off, as this graph illustrates. So if you are in a low carbon system, which a lot of kind of dry land systems are, at these lower ends of the range, from 0 up to 2%, yes, there's a good correlation. Any increase you can in soil organic matter will ultimately link with yield. But once you reach that 2% cutoff, okay, there are then other factors, other climatic and other factors at play. So we don't see a perfect, clear, linear relationship there. But at the lower end, yes, it is important as a factor for yield, not just for soil health. Okay, so here's this key message, and I'm sure you've all heard this part of me talking about some of this before, and we've touched on it now already, but really the key to building soil organic matter is roots. It is roots and not shoots. The amount of above ground residues are not a key factor. That's not what drives. Don't get me wrong, they contribute. They make a piece of the puzzle. They contribute to soil organic matter, but they're not the key driver. It is all about roots, and the more roots you can grow, the more root biomass, that will be the key to building soil organic matter. And we have lots and lots of studies, just to list them all at the bottom, that all say this, that all support this idea, that roots are really the key factor. And it's no special kind of secret behind this. It's just a fairly obvious relationship there. Roots, where are they? They're physically located down in the soil, where they can be captured, broken down, stabilized, turned into soil organic matter. All those residues on the above ground, the shoots, of course, they're above, they're on the top. It's hard, they're just physically not in close contact with the soil and the microbes. So they're not as easily captured and transferred and turned into soil organic matter. So it's not any great big secret. It's just that that's where roots are located. Now, key, however, it's not just about growing roots, it's then how they are processed by the microbes, how they are transformed in that digestion process and then turned and stabilized into soil organic matter. And so we have now also part of this nuance of this discussion, we have emerging new ideas about soil organic matter. There's a lot of new science. There's, of course, with topics like climate change and everything that's been discussed in the last decade or so, of course, there's been a lot of interest in soil organic matter science, a lot of new studies, a lot of new investigation at this. And so we have an emerging piece of knowledge that highlights that actually, when we think about what is soil organic matter, the traditional view focused very heavily on the fact that soil organic matter was made from plants. It was plant material that is decayed, that is broken down, like the roots, etc., and turned into soil organic matter. Now we understand that actually it's the microbes and them processing that plant material, those plant residues, and then as their bodies grow and die, this microbial turnover is really, really important. And actually what we call soil organic matter, stabilized soil organic matter, is actually a lot of dead microbial bodies. This is called microbial necromass. This is an interesting, one of my favorite words, microbial necromass. And that just means dead bodies, microbial waste products, microbial detritus. So their dead bodies, it turns out, make up as much as 50% of what we call soil organic matter. So this is really important because then it becomes this question of, okay, well, what do we need to do to support microbes? How do we grow more microbial biomass? And then when that dies, that will turn into this necromass. It opens up these questions. What is the best way to grow microbial biomass so that then it stabilizes and turns into necromass? So we now know that soil organic matter is a mixture. It's a combination of plant material, decaying plant material in various states of decay, as well as then the microbes and their microbial exudates and byproducts and waste products and their dead bodies. Now, in forest ecosystems, it appears that the plant material, the plant decay is a little more important. But when we talk about grasslands or agricultural lands, it's more of this kind of leans a bit more towards the microbial contribution. So this is really important. And we're going to talk more and more about microbial processes and this idea of microbial necromass. And I'm just going to read a few quotes here just from this study. And as you can see in the title, microbial necromass on the rise. There's lots and lots of studies really exploring how this relationship, this contribution of the dead microbial bodies to soil organic matter. So let me just read you a few quotes here. It is long believed that the remnants of plant material were the main components of the persistent carbon in soil. Increasing evidence has led to this paradigm shift that dead microbial mass is more of a dominant factor, particularly in the more stable fractions, the long-lasting fractions, rather than the decaying plant material. Accordingly, this evidence is shifting the research focus from the ideas of the plant contribution and humus and humic matter into these microbial contributions. Okay, sure. However, there's plenty of blind spots. There's plenty of knowledge gaps. Okay, there's the factors that contribute to this microbial necromass stabilization are still being uncovered. So there's plenty of important work still to be done. Okay, so this was one of the key studies that really supported this. This is quantitative assessment of microbial necromass. And as they suggest, it can make up, as I say, around about 50%, more than half of the soil organic matter. And in particular, it is the fungal necromass. It seems to be that the dead bodies of the fungi make a greater contribution than the dead bodies of the bacteria. So the fungal component seems to be, again, quite an important piece of that puzzle. So however we can encourage more fungi in soils, the better. Now, just to, again, break down a little bit of this nuance, on the left-hand side, this is kind of what we would call catabolism or breakdown or decomposition. Okay, this is kind of, again, how we've traditionally thought about soil organic matter. We thought that we had crop residues. They were high in lignin, high carbon to nitrogen ratio, complex material that over time had to get broken down. And so microbes would come and chew and eat and feed, and they would release very specialized enzymes. That's these little scissors. And these enzymes would cut that complex material at high in lignin, for example, cut it up and break it down into smaller components, cut that up and break it down into very small components, which then the microbes would then feed on these smaller broken down products. Microbes can't just eat that complex stuff. They need to break it down first. They digest it on the outside. We digest on the inside, of course, in our stomachs. They digest externally. They release all these enzymes or these scissors that cut it up externally, and then they absorb the resultant kind of digestion soup. They absorb those smaller byproducts. Okay, so that was the classic view of soil organic matter. We now realize that actually what is really important is those smaller pieces, those smaller components, once they get ingested into the microbes, by the microbes, what is key is the rebuilding is what's then called anabolism. It was how the microbes grow their bodies, re-stitch these together rather than breaking things down, how they re-stitch those components together and then build their bodies, build their cell walls, build their organ, build themselves. This is what's key. It is this anabolic process, this rebuilding process of how microbes grow that's actually so important. So the more we can foster this process on the right, anabolism or rebuilding, resynthesis, this is key. And it's the microbes that do this. Okay, so what about then root exudates? And here we have a little bit of this nuanced discussion, which is this difference between a little bit on the quality of the carbon. So we're all used to residues, stubble, litter, and we identify this as being really important to break down and to contribute to soil, help build soil organic matter. And of course, it's really important for soil protection and erosion control and keeping moisture in and all those other benefits that stubble brings, of course, lots and lots of benefits. But it also has to break down and ultimately help to form soil organic matter. And there's a really interesting then part of this new paradigm, new thinking around soil organic matter is to acknowledge the contribution of the root exudates, those carbon compounds that are excreted out of the root systems from living roots, of course, can be 30-odd% of the total amount of carbon that the plant is fixing through photosynthesis is pumped out and released as root exudates. Well, those root exudates are, of course, full of carbon, sugars, carbohydrates, amino acids, all sorts of things, all sorts of other biochemicals in there. And those, it turns out, are very, very efficient at feeding microbes. Microbes, if given a choice between this food on the left that is highly available, easy to digest, very rapid, very easily metabolized, very easily absorbed, very easily digested, it's very efficient for them to feed on this. Compared to that stubble, high in lignin, complex material, it's like eating a tough steak, like it takes a lot of work, right? We've got to chew, 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 chew. Microbes have got to put a lot of work and effort into breaking this down, the stubble, and feeding on it, breaking it down so that they can feed on it. Whereas they have to expend very little energy or effort to absorb all of that carbon from the root exudates. It is highly available, highly digestible, easily absorbed. So point being, if we have this discussion about, well, which one is more efficient to grow microbial biomass? Well, microbes can grow a lot more biomass a lot quicker on root exudates than they can on the residue. Residue takes time, it's complex, etc. So it's this nuance about the efficiency. And it turns out that because microbes can grow so efficiently on root exudates, therefore they can grow more quickly and grow, therefore, more microbial biomass. And growing more microbial biomass is really important because when that dies, it forms that microbial necromass, which can then get stabilized as soil organic matter. So we have this emerging acknowledgement that actually the root exudates also make a really important contribution to soil organic matter. It's not just roots. Okay, roots are definitely more important than shoots, but it appears that the exudates may be even more important still. And we have a couple of studies that have tried to untangle this and suggest what I'm talking about here. Let me just cut to the chase here. They were basically comparing how efficiently microbes grow on exudates versus more complex material. And they show that those what they call living root inputs, or those root exudates, were 2 to 13 times more efficient than litter inputs in forming soil organic matter. Some of those different qualities of soil organic matter. So, okay, we have a study that's in that direction. That's from 2018. Here we have another study from 2021, again, building on this, again, suggesting a similar trend. We found that the rhizo deposition inputs have the highest efficiency, 46%, as compared to roots or above ground inputs. So again, they're suggesting that these highly available root exudates, these very fresh rhizo deposits from living roots, are most efficiently captured by the microbes, turned into microbial biomass. And of course, the efficiency, or how easily we can do this, is really important, because the more inefficient we are with that transfer into microbial biomass is that we're opening the window for more of those losses. Remember I said most of the residue that comes in ultimately goes off as CO2, 3/4 or so goes off as CO2. That's what we're talking about there. The more efficient, or the more quicker you can transfer the carbon into the microbial body, the less chance there is of it all kind of being oxidized or being lost. Okay, so the efficiency of that conversion is really important. So, okay, we have a couple of studies really pointing in this direction that the exudates are also a really important piece of this contribution, which is why it's so good that we have, for example, the soil health principle, you know, this idea of living roots 24/7, etc. It is such an important part of the soil health principles. I really support that one very much so. We have now the emerging kind of evidence to really support it, that yes, it's living roots that and those exudates that are so key in terms of this carbon use efficiency. So I indeed glad to see we're having, you know, more strategies and more discussion about whatever post-harvest cover crops and trying to keep winter cover and all these things on. There's good evidence to support this. Okay, so let's now also get into some nitty-gritty. Everybody relax, settle into your seats. We're going to stay on this slide for a long time. I'm going to probably talk on this slide for a good 10 minutes or something. There's a lot here to say. So here we're going to talk about this point about different quality of soil organic matter, different types of soil organic matter. And we're going to talk about then how these formed, the different kind of pathways in which these different types of soil organic matter get stabilized and ultimately form. Okay, so there's two kind of main groups, or two different types, let's say. One is called particulate organic matter, and one is called mineral associated organic matter. Okay, so these are two different qualities of, when we talk about soil organic matter, these are two different qualities that exist under that broad heading. And we're going to talk a little bit about these two and just some of the differences and nuances between them. So let's start with, on the left, particulate organic matter. What that really is referring to is what I've kind of touched on a little bit here just earlier, this kind of nuance of this slide, talking about, you can see on the left here, it's material that's it's a continuum. It's plant material that's in various states of decay. It's in this breakdown process. There's some complex stuff, there's some middle ground stuff, there's some stuff that's more broken down. It's a continuum of decay, a process of that breakdown. And so what we're seeing is that plant material, so those residues, comes into the soil, and that's what happens. It starts getting cut up by those enzymes. So it's in a continuum, it's in these various kind of states of decay. And that's really what we would call particulate organic matter. It is plant material, and it is that plant material that is in that process of being broken down and getting smaller and smaller and smaller, being attacked by those enzymes, etc. Okay, so that's on one side. Now, the material that comes from, that particulate organic matter, as I say, is your crop residues, stubbles, roots, all of those things that we were also just talking about. And that material, therefore, it's not very, very stable in the soil. It's kind of going through a breakdown process. It hasn't yet been consumed by a microbe. Again, this is this kind of nuance here. It's getting attacked by those enzymes, attacked by the scissors. It's not yet small enough where it then gets absorbed by the microbes. It hasn't been absorbed or ingested by the microbe yet. It's still being externally attacked, still getting kind of broken down. And that basically means that it's a little less stable in the soil. It means that it's getting cycled, it's getting turned around, it's getting used. It hasn't been stabilized in the soil as yet. So it's called, you know, this fast cycling you can see here, lifetime 1 to 50 odd years. So it's the fraction of carbon that's continually being cycled, getting used, but breaking down in these various states of decay. The key point, again, just to emphasize, and I'm repeating myself a little here, but it'll all come together. The key point about this particulate organic matter is that it has not yet maybe got small enough that it is then ingested by the microbe and taken up by the microbe, where then it is influenced by these anabolic processes. Hasn't been influenced by a microbe yet, just externally, just those microbial enzymes that are breaking it down and cutting it up. So that's a fraction of soil organic matter called particulate organic matter. Now, on the other side, this is the key point, once that particulate organic matter gets small enough, once it is ingested by the microbe and becomes microbial biomass, now we are opening up the other pathway. Now that carbon got into the microbe, now that microbe grows and dies and grows and dies and does its thing and feeds and lives happily and et cetera, et cetera. And when that microbe dies, that microbial body, the cell walls, the components of that microbial necromass, these are very able to be then preserved and stabilized in the soil. And the way in which that happens is those microbial dead bodies, particularly, they're attracted to the mineral surfaces of the soil. So the sands, the silts, the clays. And of course, the more clay material, the more mineral surfaces you have in your soil, the more potential you have to form this mineral-associated organic matter. So it's simply organic matter that's stuck to mineral surfaces, stuck to the soil colloids. So of course, lighter sandy soils, they have less mineral surfaces, so they have less potential to form this minerally associated organic matter, this stuck and stabilized organic matter that's stuck to those mineral surfaces. Whereas a medium soil or a heavy textured soil, clay soil, etc., they have a much greater potential. So the key kind of nuance or difference between these then two types is that once it gets stuck to the mineral surfaces, it becomes very stable. So then it's called slow cycling, has a much longer lifetime, potentially up to 1,000 years, for example. So it gets stuck and stabilized. Now, there's a little differences between the carbon to nitrogen ratio of these two materials. As you can see here, the particulate matter, it's made up of a bit more carbon with a little bit of nitrogen. Whereas to form mineral-associated organic matter, the more stable fraction, you do need a little bit more nitrogen. It contains more nitrogen in it. That's because the microbes themselves need a bit more nitrogen. Their dead bodies then contain more of that nitrogen. So it's a little bit more of a nutrient-rich fraction of soil organic matter. It's there for being used, drawn upon for a source of nutrition. A bit more nutrient-rich in that sense. But as I say, it's a little bit more locked up. It's a little bit more stabilized. It's a little bit harder to access equally. And the key point to then understand about these two different types is the pathways in which they're taking and the quality of the organic material, the residue, the quality of the plant residue that contributes to these two pathways. So what we see is that on the left-hand side, what we would call low quality litter. So this is stuff that has a very high carbon to nitrogen ratio, very wide carbon. This is the lignin, your stubbles, your very complex. This is the low quality stuff. Because that is big and complex and difficult to break down, then it has, again, this point about the lower carbon use efficiency. It's harder for the microbes to just simply ingest it. So you'll find that poor quality litter or high carbon to nitrogen ratio litter, it generally then goes down this pathway of forming more of this particulate organic matter because it's getting attacked by those enzymes, getting broken up in that continuum of decay. Whereas better quality litter, and here I'm now referring to, for example, legume litter or litter that has a lower carbon to nitrogen ratio, so more nitrogen rich litter. So for example, legume litter. Because it has a lower carbon to nitrogen ratio, as you all, I'm sure, know, that means that microbes can feed on it more easy. It's easier for them to digest. They don't need to release all of those scissors, all of those enzymes to break or put all that work, all of that, because this takes microbial energy. This takes their metabolic energy to produce all of those enzymes. It's kind of wasting their energy, so to speak. They are using metabolic energy to synthesize those enzymes to attack that litter. Whereas if they didn't have to waste their energy to produce those, they could just absorb the residues more rapidly and not have to expend all of that metabolic energy to break it down first. So as you all know, legume litter, it breaks down very quickly. This is because it has a lower carbon to nitrogen ratio. There's more nitrogen in it. It's more easily digestible. Okay, so the very fact that it is more easily digested by the microbes means that it is entering their body. That carbon, that quality of carbon is entering the microbial body. And therefore, it's more likely to go down the pathway of forming this mineral-associated organic matter. Okay, it's going to be incorporated into the microbial body where it will then die, form that necromass, as we mentioned, and then get stuck onto soil surfaces and soil particles, etc. Okay, so it's that same nuance that we kind of talked about here, where we said, okay, root exudates are a better quality. Microbes can very easily digest this. This takes a lot of work. So that kind of nuance. So the same deal with exudates being kind of more preferable or having a greater efficiency. Similarly with the quality of the litter. The legume litters, they break down more easily. They're more likely to go down this pathway of being ingested by the microbe and forming this mineral associated. Okay, so that's a kind of a nuance. Now, and you know, I know a lot of you would say, well, we don't like too many pulses in the rotation. That, you know, doesn't give us the soil cover. That litter breaks down too quick. You know, we view this as almost a negative thing because we're not getting that soil protection. And of course, that's also really important. We want soil protection. We want stubble. We want residues for all of those soil protection, erosion, and moisture conservation for all those very important reasons. But just so you're aware, part of the reality is that as that legume litter is decaying so quickly, that we kind of view that as a negative thing. What that's also doing is breaking down quickly, but also forming a better quality of soil organic matter, this more stable fraction, this mineral associated. So although it's visually to our eyes disappearing quickly, it is forming a better quality, a more stable fraction of soil organic matter. And as you all know, soil organic matter is really important to hold moisture in the soil, not just protect it from the top, but actually store it within the soil as well. So I think there's a little bit of nuance. I know legumes sometimes have a little bit of a bad rap. I think there's still some space for them. I think one of the very interesting things about the intercropping phenomenon that we're seeing, I think there's a lot of really interesting and good opportunities here, and we'll talk about this at the end of this talk, is to have a little bit of cereal stubble and legume stubble together. I think monocultural pulses are particularly a problem. And then, of course, you're left with no soil cover. But having a little bit of cereal and legume together means you've got a little bit of best of both worlds there. Yes, the pulse will degrade quicker, form that good quality of soil organic matter, and then you've still got the cereal stubble there for soil protection and moisture conservation, all those other benefits that you also want. So I think I'm really enthused to see some of the legume cereal intercropping kind of unfold and happening at the moment. It's great to see. And I think there's going to be some really interesting benefits for the quality of the soil organic matter that you're forming from those systems. Okay, so that's a kind of a bit of a deep dive just to point out these two different types. I hope I've explained that. I did repeat myself a little bit here or there. I hope I've kind of explained that nuance. Again, key point just being that the better the quality, the material that comes into the soil, the more easily the microbes can digest it and feed on it. And that ultimately leads to more microbial biomass production, more microbial necromass production, and then stable soil organic matter. Now, I should also say that although this is considered the more stable fraction, it's more permanent, so to speak. And there's no such thing as ever real permanence, but more stable and more permanent. I should also say that that's not to detract from particulate organic matter. It's still very important. It's still making a very important contribution to the overall soil health and soil function and the soil organic matter pools. It's just that they're different. They're just two different types. It's not necessarily to say that one's more important than the other. It's not to say that the mineral associated is more important just because it's the more stable form. They're both important. They're just a little different, and that's the key thing there. Just to point out. Now, I should also say that particulate organic matter, bits of stubble in various states of decay, it can also be stabilized into soil organic matter, but that's mainly through physical processes, through aggregation. So if we build aggregates, if the microbes and the fungal hyphae are growing through soil, pushing soil particles together and aggregating the soil, within that aggregate, you can have low quality material like particulate organic matter. You can have that physically stabilized. It's physically stabilized in the aggregate. Okay, so again, particulate organic matter, it can still be stabilized in the soil, but it's dependent on aggregate formation. And of course, if that aggregate breaks down, or if we disturb the soil, cultivate the soil, for example, and we break apart that aggregate, then that particulate organic matter is very vulnerable to being lost, to being oxidized off and broken down. Whereas the mineral associated one, much less so, because it's tightly chemically bound to those mineral surfaces, to those colloids. But okay, just to point out that the particulate organic matter can be stabilized physically through aggregates. And so again, this is really important. So I tease apart these two different types because they're very different. They form in different ways and they have different qualities. And we have this discussion and this debate about do we reach soil carbon saturation? Is there a top end of how much soil organic carbon we can build that we can sequester in soils? And of course, if we break apart the nuance of those two different types, we see two very different pictures here. So on the left-hand side, we're looking at this mineral-associated organic matter. And of course, because mineral-associated organic matter needs mineral surfaces for it to form, it does saturate. Once you have filled your sandy soil, all those mineral surfaces on, if you've got a sandy soil, once it's full, it's full. You've got no more mineral surfaces to occupy. You've attached all the possible carbon that you can to those mineral surfaces, and you cannot keep sequestering more carbon, more minerally associated carbon in that soil. Now, of course, a medium soil or a clay soil is going to have a higher threshold. It's going to have more mineral surfaces in it. But either way, once they are all covered and stuck to and saturated with carbon, we can't keep building more mineral-associated organic matter. It has saturated. So you can see that it plateaus off. Okay, we can't just keep going. We've filled up the tank, so to speak, it's full. Okay, there's no more that we can fit in there because all of those mineral sites are colonized, are occupied with carbon. So it does saturate, yes. However, the particulate organic matter, as you can see on the right-hand side, it does not saturate. It keeps just going and going and going. Because of course, it's not dependent on those mineral surfaces for it to form. It's just plant material in those various states of decay, in that continuum of decay. So yes, it can just accumulate and accumulate and accumulate. And that is kind of what we see in a lot of the forest type ecosystems, a lot of the boreal, evergreen, pine type systems. They've just got lots and lots and lots of dead leaves, litter, just accumulating. And that's kind of why we see lots and lots of carbon. So sure, it is a lower quality fraction, as I talked about, particulate organic matter. Sure, it's lower quality, it's less stable, but as you can see, there's more potential to build more of it.
Sorry to interrupt, Joel. We have two questions from our virtual viewers.
Okay.
One
Sure.
of them is, can soil organic matter become hydrophobic? If yes, how can this be overcome?
Soils and non-wetting soils, yeah, and these are a real challenge. I don't have an easy answer to this question. It is a very common problem in many parts of Australia. It's a big problem, particularly in a lot of sandy soils. And it's to do with the residue, crop residue, as it breaks down and goes through some of that decomposition. There can be some fats, oils, some waxes, some of the waxes in the plant residue, in the litter, which can then not fully break down and then kind of stick to soil particles and then make the soils very repellent, hydrophobic. So yes, this is a big problem, and I don't have an easy answer for the listener in terms of overcoming that. There's a lot of researchers, for example, in Australia, there's a lot of money looking into this at the moment and trying to find those answers. But they're exploring things like trying to find certain microbes that will digest those waxes better and break them down more effectively. That seems to be one avenue. But otherwise, yeah, there's no easy answer to that question. Sorry, I don't have a good answer for that one. Tanya seems in the middle of something. Do you want to do that second question now, Tanya, or should we come back to it?
The second one is, are the mineral surfaces in a sandy soil likely to be saturated with MAOM? If so, what happens to additional necromass?
Okay, yeah, so there's a good question. So yes, a sandy soil will rapidly get saturated, and because it only has a very small amount of those mineral surfaces, hence it's a sandy soil. So they will rapidly get full and saturated. Then in terms of the any remaining microbial necromass, it then can only be stabilized through that physical pathway that I was also talking about, through the aggregates. So it can get, microbial necromass can still be captured and trapped physically within an aggregate. So it can be stabilized that way. And also just to point out that microbial necromass itself is not permanent. It can also be broken down. Actually, living microbes will feed on microbial necromass. So some of those microbial dead bodies can also be a source of nutrition to be cycled. So it is also used as a food source for the new next generation of the living microbes, the current microbial biomass. They will also feed on necromass. So that's what's going to happen to necromass. If it's not adhered to mineral surfaces, once that's saturated, whatever's left can be physically stabilized through aggregates, or it then just becomes further broken down and constantly cycled and consumed by the remaining living microbes. Do we want to take, is there any other maybe questions from the floor while we're having quick question time? Anyone else? Yep. Mm-hmm. Yes. Okay. Just for the audience at home, the question was, I mentioned that fungal necromass makes a more important contribution to soil organic matter. So the question was, what practices can we do to support fungi and the development and growth of fungi? We'll talk a little bit about this afternoon. It'll be part of that discussion, but a quick answer now is, so moving away from tillage is number one. That's the big one. Every time we cultivate the soil, we do break up those fungal hyphae, those networks. So that's number one. Trying to use less fungicides, of course, wherever we can. Fungi are often somewhat sensitive to even some of our herbicides and things as well. So wherever we can eliminate some of our pesticide use is always going to be beneficial. And then really it's about encouraging them through feeding them. And that could be applying, and this is what we'll talk about this afternoon, but applying certain inputs or fungal foods to kind of feed them, things like humic acids or fish kind of hydrolysates or these kinds of things. But also more simply, just again, living roots, keeping living roots, keeping the soil covered. If you've got living roots, you've got root exudates, and those root exudates are always hands down going to be the preferred food source for microbes. So by encouraging living roots and good fungal associating plants, legumes are generally also, they're quite good fungal associators, so they're very good for this. But also then there's a fair few studies that would suggest that diversity, cocktail cover crops, or at least mixed species, getting a bit more diversity into the system. This also encourages a better fungal activity as compared to monocultures as a general rule as well. And we'll talk a bit about that this afternoon. Okay. All right, so let's move on to the question. We've got through all of that kind of background. I took a while to get through all of that, so hopefully we'll move through these next parts a bit quicker too. Okay, so what's the answer then? What does nitrogen do? Does it burn or does it build soil organic matter? And the answer is both. And it depends, and it's context dependent, and there's all sorts of kind of variables that come into play here. So there's no easy answer to this one. So we have like a real mixture of studies on this. The evidence is very, very mixed. We have plenty of studies that show that nitrogen inputs can increase soil organic matter. They can build soil organic matter, and that is through greater residue input into the soil. Nitrogen helps to grow more biomass, as you all know. That means there's simply more food coming into the soil, more residue coming in, which can potentially build soil organic matter. As I touched on at the beginning, that's not a linear relationship. However, just because litter is coming into the soil doesn't mean it stays there. It's again about the quality of that litter. How easily digestible by the microbes is it? There's a little bit of nuance to that whole discussion, which we were going through. So we have studies that say, yes, it can help to build soil organic matter through more biomass production, more residue input into the soil. But we also have studies that say it can decrease soil organic matter. And this is because of increased mineralization, so nitrogen mining. Okay, I'm going to talk about this, but that nitrogen can stimulate the breakdown of the soil organic matter, can kind of release it and break it down, so kind of burning it. So we have studies that support that, and we have plenty of studies that also say that there's kind of no effect, it's neutral. So it's a total mixed bag, and it seems to be, there's no easy answer. So, it depends. And it's a lot of kind of context dependent. Here's just some examples. I'm just kind of, we're not going to go through these in detail. I'm going to just cherry pick the kind of the core of the answer here. But, just some examples. Maximum storage when crops are optimally fertilized. synthetic nitrogen depleting soil carbon. Here we have nitrogen fertilizer suppressing that mineralization of organic matter, so it's helping to stabilize it. The myth of nitrogen fertilizer for soil carbon sequestration, long-term fertilizer gradient has little effect on soil organic matter. I mean, it's just a total mixed bag of evidence. And so let's try and have a little unpack of this as to kind of why and maybe what some of these variables and factors are. Now, we've touched on this a little bit, and I'm going to go deeper into this. Here's the first example that we heard about already. I pointed out that with an increase of fertilizer intensity, and this is true particularly of nitrogen, yes, it seems to drive above ground biomass. It drives yield, yes. It doesn't necessarily drive root biomass and root production. Okay, so that's part of the nuance. It is ultimately all about those roots and root exudates that are so key to building soil organic matter. So there's that part of the effect. And here we have another graph illustrating the same point. This is a combination of 3 separate studies kind of summarized into the one graph. And we're looking at increasing nitrogen fertilization rates across the bottom. As we increase fertilization rates, nitrogen fertilization, we're looking at the below ground carbon allocation. How much carbon does the plant allocate to below ground, to the roots? And you can see here that as we increase nitrogen fertilization, we have a decline in the amount of carbon that the plants will allocate below ground. Okay, so they're growing less roots. The more we nitrogen fertilize, the plants will grow less roots. They'll grow more shoots, but not more roots. Okay, so there we have another piece of evidence pointing towards this nuance of roots, that nitrogen is not so good for roots. And I think this is really my key answer. This is a really good study. This was from also recently, just last year, 2021. And I think this paper we've lost, oh, we're back. We lost power again, Tanya, but we're back. Okay. And I think this, for me, this is what it boils down to. I think this is the key kind of take-home message for today's topic. And we're going to dive deeper into this still, but I think this is kind of the nice, kind of easy summary slide and the key take-home message. Insufficient and excessive nitrogen fertilizer input reduces maize root mass across soil types. Okay, so root mass was maximized near the optimum agronomic rate. This is N fertilizer. I'm just reading a couple of highlights here. Affected the roots traits only in the top 30 centimeters. Soil texture also had an effect. And the root to shoot ratio decreased with increasing levels. Okay, so let me just come to these pictures and we'll explain it perfectly here. So really the answer is the green dots. We're looking at the yield. We're looking at the yield here. So you can see as we increase nitrogen, yield goes up to a certain plateau. Okay, so yield plateaus off, and you can see that same here. These are two different years. Now, what you can see is the red line, the roots. This is the root biomass production. And so you can see that when we have insufficient nitrogen, root biomass is compromised. It's limited, and it will increase with increasing nitrogen rate up until a point. And when we then increase nitrogen further, we can see that the root biomass then declines. And same thing happened less intensively, but also in this other year. So for me, I think this is the answer. If nitrogen is limiting, of course, it's deficient. Therefore, crop production, biomass production is also going to be limiting. And that includes root growth. As we increase nitrogen, of course, we can optimize biomass production. But that is up until that point. Any surplus nitrogen that we apply begins to have a negative impact on, specifically on root biomass, on root biomass production, kind of really supporting also supporting this slide. And I think that's the key answer. Too much nitrogen will reduce root biomass production. It is roots and not shoots that ultimately build soil organic matter. And so this is the key piece of this discussion, is the nuance of how nitrogen affects that below ground carbon allocation, that root biomass production. So the simple answer is you just have to try and optimize your nitrogen application. And if you optimize that, then you're good. But of course, that's the tricky question. What is my optimum amount of nitrogen? It's different every year. Is it going to be a dry year? Is it a wet year? The different soil types, the different contexts? What is the optimum amount of nitrogen? That's the hard bit to answer. But that is, I'll acknowledge that's the tricky part. But the answer more broadly to optimize root production and therefore optimize soil carbon is just try and get that nitrogen optimized. Yeah, that question there. Yeah, great question. Excellent question. And that's why I'm going to move on very shortly here and talk about soil organic nitrogen, which is a topic that we don't have enough conversation on. And yeah, so these studies, as one of the negatives or problems with them, I would say, yes, these are all being done with synthetic nitrogen. Now, as we're going to move on and talk about soil organic nitrogen sources of fertility inputs and just the way in which soil organic nitrogen behaves in soils. We're going to unpack more of this very soon. And it's a very different answer. It is very conducive to building soil organic matter. You don't have all of this kind of nuance and variation here. Soil organic fractions are very good for building a soil organic carbon, soil organic matter. Yeah, that was a great question. Yeah, that's a good question too. So yes, it's a trade-off. The more carbon we allocate down to the roots, the less we are leaving above ground for yield. It is a trade-off. There's a reason that we have bred for yield over the past 50 odd years, is because we flick that switch into more allocation above ground. That's how we got more yield, by doing that at the compromise of the trade-off of roots. Hence, we've seen these nuance of soil organic matter declines. So if we start breeding for roots and going down that direction, yes, it will come at a trade-off to yield, to above ground biomass production. However, then it becomes that nuanced discussion of, well, is that necessarily a bad thing? Is that economically necessarily going to hurt you? And it may not necessarily always. So a bigger root system also means that it is exploring more soil volume, accessing greater soil moisture, accessing greater soil nutrient reserves. So there's the potential to lower your input costs, lower your fertilizer, because your bigger root systems are accessing more soil volume, leveraging more free fertility from the soil. So there's potentially a cost saving there to offset the yield loss. And additionally, then particularly, I think in dryland environments, it could be a price worth paying in the short term. For example, if we maybe grew 5 years, if we went down this pathway for 5 years, grew lots and lots of roots, okay, you took maybe a slight hit on the yield for a couple of years, but you could offset that with fertilizer prices. But if you did that for a good 5 years and you really built soil organic matter better, in a dryland system, if you then hold on to more soil moisture, that could then also pay dividends, and you can catch up on that economic loss later on with a better functioning system of better water holding and better water use efficiencies. So that's a tricky question too, but that's my thoughts. Yes, okay. I'm going to talk a little bit about carbon-based stabilizers as we go along, things like humic and fulvic acids, and I think they can play a really good role in that carbon in a very user-friendly, microbe-friendly way to capture and bind to stabilized nitrogen and also feed microbes at the same time. So I think they're very good options, and I'll talk a bit about those. But even some of our ureas and nitrification inhibitors, yeah, I think they have a place for sure. I think anything we can do to minimize nitrogen losses and therefore be more efficient with the amount of fertilizer that we use, then yes, I think all tools should be on the table. And I think some of the nitrogen inhibitors can be a part of that for sure. Yeah. Tanya.
From online, interesting slide comparing the roots of Eichhorn to a modern durum. Does the difference in roots translate directly to a difference in nutrient density in the harvested grains?
Oh, that's a good question. For sure, the traditional varieties, all of those ancient varieties, are more nutrient dense, yes. Whether or not that is directly because of those root systems and those bigger root systems, I don't know. I'm not sure if that is necessarily the exact mechanism. But certainly the evidence is very clear that when we have this kind of discussion about nutrient density, there's quite a few studies that really show this, that it was at the advent again of the Green Revolution, of modern plant breeding, of higher yielding plants, where we did see nutrient declines. It is the modern varieties that have led to a nutrient decline. And that's because they're more higher yielding, and therefore the nutrients in them become more diluted because they're higher yielding. So that's part of the reason that they are less nutrient dense, is simply because the nutrients that are there are diluted through a bigger yield. Now, whether or not that directly correlates back to the root system side of things, I'm sure it's a factor and at play there, possibly. So maybe, but certainly it's clear that some of the older varieties are more nutrient dense. And I think there's a lot of discussion about nutrient density and how we can improve it and tweak it. And I think it's all really good and valuable, important conversation to be having. But I would say for sure, one of the easiest ways to do that is just grow some of those older varieties. They're by default going to be more nutrient rich. Okay, right. So that's the simple answer. It's about optimizing nitrogen, and I think it just makes sense. I've showed you the evidence here. It's because of the impact on roots and below ground carbon allocation and impact on root biomass production, and that is why it then has this nuanced effect. Too little can be bad. Too much can be bad. We want to try and get that Goldilocks principle in the middle ground there. So just very quickly, just to point out this idea then of, well, what about how does nitrogen, how does it kind of burn soil organic matter? What are the kind of the processes here? I just want to visually try and illustrate this here, just so everyone's clear on this, because we will talk about it a little bit later. Just picture the brown half here is kind of soil organic matter, and acknowledging that soil organic matter is not just carbon. It's not just made of carbon. Soil organic matter is a mixture of things. It's also got nitrogen. It's also got phosphorus. It's also got sulfur. These are the main, and plus plenty of other nutrients and micronutrients and things. They're all embedded in that. Again, this point that soil organic matter is a mixture of lots of different things, different types, different qualities of soil organic matter. So just acknowledging that soil organic matter itself is made up mostly of carbon, but there are these other nutrient components as well, nitrogen, phos, and sulfur. And when we apply nitrogen, if we have fresh applications of nitrogen then in the soil water, in the soil system here, and we have all of our biology, our soil microbiology here growing, of course, they're going to be particularly crowding around the soil organic matter because this is where they live on and they like to feed on it and live in and around it, etc. So they're here closely associated with physically with the soil organic matter. And when we have a nutrient enrichment, nitrogen, a lot more nitrogen in the system, well, of course, these microbes get that resource. They eat at the table first. They're going to also be absorbing that nitrogen. They're going to be using that nitrogen, and they're going to begin growing their microbial biomass. It's a nutrient for them too. They need all the micro and macronutrients for their bodies too, just like plants do, just like we do. And of course, their bodies are made up of nitrogen and carbon. Do you want me to jump to you now, or can I finish this point? Finish?
Yeah, you can finish.
Yes, okay, so their body, the microbial bodies are made up of a carbon to nitrogen ratio. The bacteria, theirs is about 5:1. Fungi, theirs is around about 20:1. So they, to grow their microbial biomass, to keep that in balance, fungi need about 20 carbons for every 1 nitrogen, and bacteria need about 5 carbons for every 1 nitrogen. So as they get enriched by nitrogen, as they start to feed and get that resource and they start to multiply and grow and also subdivide, they need to optimize their carbon to nitrogen ratio in their bodies. So they will go and mine the carbon from soil organic matter. If we enrich with nitrogen, they need to keep their bodies balanced. So they'll go and start stealing the carbon mining, as would be called, mining the soil organic matter specifically for the carbon. That's what they're going to be chasing, to then combine carbon and nitrogen in their bodies and optimize their ratio. So that's the mechanism by which we where nitrogen can induce carbon losses, is because we are unlocking or we are mining the carbon out of the soil organic matter. But that also goes 2 ways, whereas when we have equally a carbon enrichment in the system, as we start to have a lot of crop residue, as it starts to break down, for example, and it's also we're having an enrichment of carbon in the system. And although that's a good thing, we always want more carbon in the soil, but I'm just pointing this out to you. We have the same phenomenon that's going to happen. Microbes are now being enriched by carbon, but they still need to optimize the ratio of their body 5-to-1 or 20-to-1. So they will also still mine soil organic matter, but they will be mining the nitrogen. They're going to be then specifically releasing specialist scissors, specialist enzymes that are unique to grab onto nitrogen within the organic matter and cut that out and get that nitrogen. So we will see nitrogen mining from soil organic matter, even where we have carbon enrichment. Okay, so I'm just pointing out that simply just putting more carbon in, that's all good, but it's still this process of mining or accessing and unlocking the nutrients in the soil organic matter. And I say that just to point out that that's okay. Soil organic matter is there to be used. We have this kind of idea that we must sequester, we must build. In order for soil organic matter to be functional, to be useful, it has to be being broken down. It is the process of being cycled, being broken down, rebuilt, broken down, rebuilt. That's what makes it functional. Just having it sequestered and stable and doing nothing isn't necessarily so beneficial. It has to be active, it has to be cycling. So there's an element of this carbon cycling, nutrient cycling, nitrogen cycling, carbon cycling, that is perfectly healthy and perfectly what we want. Organic matter is there to be used and then to be also rebuilt, replenished. Has Tanya gone out of the room? I think she has. Okay. So that's just the nuance of that mechanism there. Now, what about the effects of nitrogen on biology? I mean, this question that we get asked 100 times, what are the effects of pesticides on soil microbes? What are the effects of fertilizers, synthetic fertilizers on microbes? If I was given $1 every time I was asked these questions, I'd be a millionaire. So let's have a look at what we think about then some of the effects of that nitrogen. It's not just about soil organic matter, though that's a really important discussion. What about the effects on the microbes themselves? So here we have a study looking at this. Again, this is one of these meta-analysis, so a study that looked at lots of other studies and brought all of those various studies together and reanalyzed them and re-summarized them into one study. So we can see we are conducting a meta-analysis here of 1,248 kind of observations from 120 different studies at 125 different sites that cover many different biomes around the world. The results show that nitrogen enrichment significantly decreased microbial biomass carbon, as well as mycorrhizal fungi across all studies. Our findings highlight that nitrogen enrichment decreased the soil microbial biomass and also showed significant effects on the microbial enzymes, those various enzymes. We can also measure those scissors, those enzymes that I was talking about before. We can measure those as indicators of soil microbial function and activity. So we can see significant changes in those enzymes across terrestrial ecosystems with more pronounced effects observed with increasing nitrogen rate and the duration. Okay, so here we have the summary graph here. You can see that microbial, if we're kind of this way, it means that nitrogen is decreasing those things. So you can see microbial biomass is significantly lower, mycorrhizal fungi also particularly lower, and some of the other enzymes influenced. Okay, so here I'm just saying that nitrogen fertilizer does appear to have a negative effect on microbial biomass. Now, it's not to say always, indeed small amounts can induce positive effects, etc. It's nuanced, but okay, from this study of reanalyzing lots of other studies, the broad consensus is that, or the broad finding is that that nitrogen fertilizer has a negative effect on microbial biomass production. Now, that's really important because what happens to biomass, microbial biomass, when it dies? It forms that microbial necromass. So by inducing negative effects on microbial biomass, we are obviously lowering the potential to form microbial necromass. And as we now know, that necromass is such an important component of soil organic matter as well. So, so it's not just about those overall effects on soil organic matter. It appears we should also be thinking a bit more broadly about some of the other interactions as well. And I put this one up here just again to show that nuance, that it's not always black and white, but mineral nitrogen input decreases microbial biomass in soils under grasslands, but not in annual crops. Okay, so this particular study suggested there was a bit of nuance there where nitrogen decreased microbial biomass by about 12% in grasslands. However, in an annual cropping system, it seems to increase by around about 13% odd. where the microbes benefited from those higher residue inputs coming into the system. So again, that's just, again, one study, whereas this previous one is reviewing lots and lots of other studies. So it's kind of broader in its findings, but I'm just highlighting to you, like always, it's a bit of a nuance and context kind of dependent system, context kind of dependent scenario. Okay. So here's this question about then what form of nitrogen, this nuance about what were some of the studies doing there. Of course, they were using synthetic nitrogen to try and answer this question. And, fine, that's what a lot of our agricultural production systems are based on. We do use nitrogen fertilizer, so trying to untangle those effects is important, yes. But also just to say we should be having some conversation about the organic fractions of nitrogen as well, which is what we will kind of talk about now. Okay, so when I talk about organic nitrogen, what am I referring to here? Now, just for your awareness, that most of the nitrogen in soils, above 90% of the total nitrogen in soils is organic nitrogen. Now, there's temporary points in time where that's not the case. Straight after you apply nitrogen fertilizer, of course, you're going to have a very big influx of inorganic nitrogen at that time. So at these kind of key points in time, it's not always the case, but once that integrates into the system, etc., 90+% of the nitrogen in soils is organic matter, is embedded within organic matter, or is in these organic fractions and forms. So it's a major part of soil nitrogen, and yet it doesn't, again, this organic nitrogen doesn't seem to be a topic that we discuss with much interest and fervor as we do inorganic nitrogen. So let's dive a bit deeper into this organic aspect, because it is really important. Now, what is soil organic nitrogen? It's things like proteins, peptides, which are kind of small proteins or little small chains of amino acids, and something called amino sugars. These are components of the microbial cell walls and that microbial bodies and that kind of thing. So there's various different organic components that make up what is organic nitrogen. And I mean, what is it at the end of the day? Organic nitrogen is nitrogen and carbon. It's nitrogen and carbon embedded together. It's forms of nitrogen that have carbon attached, for example, other carbon kind of components. Here we have inorganic nitrogen, just the nutrient on its own, but once it's entangled or embedded as an amino acid or a protein chain or embedded with pieces of carbon, it basically forms what we would call organic nitrogen. Now, it turns out that plants do not just take up nitrate and ammonium. Again, classic plant nutrition thinking has dominated heavily on the role of nitrate nitrogen, the role of ammonium nitrogen as the main and primary sources of nitrogen nutrition for plant growth. And of course, yes, they are very important. Yes. However, plants can take up directly amino acids. They can absorb organic nitrogen like amino acids directly. They can even absorb some of these peptides, these kind of small proteins. They can also absorb them through the roots. They can use these organic sources of nitrogen for plant nutrition, for plant growth and metabolism. And it turns out that these organic fractions are very beneficial for plant growth, which we will unravel why. Now, there's also this nuance about differences between ammonium and nitrate, but the key kind of message here is just to say that generally we find that the ammonium form encourages more root growth, more root biomass production, and that's because when ammonium is taken up by the plant, by the root system, that ammonium is usually converted in the root system. It's converted from ammonium into amino acids, into proteins. It's processed in the plant, in the roots. So therefore, it generally encourages more root production. Whereas nitrate, when it is absorbed, it goes up into the leaf, where then it is converted up in the leaf. So generally, nitrates then encourage more biomass above ground, and ammonium encourages more biomass below ground. And the other point about that is that organic forms of nitrogen are the same. They sit on this side. So when we use things like urea, but also then those amino acids and all these organic fractions, when they are taken up by the plant, generally they encourage more root production versus shoot production. Okay, so again, if roots are so key for building soil organic matter and all of that kind of conversation we had, well, it turns out that the organic sources of nitrogen tend to encourage more root production is the take-home message. Now, the key point about organic forms of nitrogen is that they're not just nitrogen, they are nitrogen and carbon, okay, as I was pointing out just before. And that carbon that comes with the nitrogen is very beneficial for plant growth. It can be used for plant growth, for photosynthesis, for plant metabolism. What does the plant do with photosynthesis? It's trying to get carbon out of the air to grow its body. Well, when we feed the plant organic nitrogen, which is nitrogen and carbon, we're feeding the plants free carbon. That therefore the plant doesn't have to use photosynthesis to get carbon from the air. We're giving it directly through the amino acid or through the protein, for example. So here's a study that was then looking at the efficiency of inorganic versus organic forms of nitrogen and looking at how they are different. So the chemical form of nitrogen taken up, whether inorganic, such as nitrates, for example, or organic, such as amino acids, may significantly influence plant shoot and root growth and nitrogen use efficiencies. I've got a slide next that's going to elaborate on this. The key point is that the carbon cost of assimilating organic forms of nitrogen into proteins is lower than that of inorganic. So when the plant takes up inorganic nitrogen, it has to expend more energy to convert that into proteins than it does by taking up amino acids. They are easier to turn into proteins. They are metabolically more efficient. And it is the carbon bonus, that free carbon, that makes all the difference. Okay, so I've got this slide here. This will summarize the key point I'm trying to make. So we're doing basically some carbon economics here. And we're looking at different carbon, nitrogen sources, nitrate, ammonium, and 2 amino acids. And we're looking at the carbon cost. Now, what does the plant want? The plant, at the end of the day, it wants proteins. It doesn't want nitrate, it doesn't want ammonium, it doesn't really want amino acids, it wants proteins. The plant has to build those proteins from all of these raw ingredients. So let's start with these two, nitrate and ammonium. There is just no carbon there. It's just inorganic nitrogen. And so there is a carbon cost. The plant has to photosynthesize, take in carbon, combine that carbon with the inorganic nitrogen source to then build those amino acids and then build those proteins. So there is a carbon cost associated with using the inorganic sources. And therefore our final overall carbon cost is the same. There's no carbon bonus here as compared to amino acids. These two, of course, this is nitrogen and carbon embedded in there. There's free carbon in there. So yes, there's still carbon cost. There's a metabolic cost to use these, to take them up, to process them. However, there is a rebate. There's a free carbon bonus. There's carbon embedded within there. So we are saving carbon. We're getting this rebate through that amino acid. That means our overall final net costs means that the organic forms, these amino acids, are more efficient for plant growth and for plant metabolism because we are providing free carbon, and therefore the overall photosynthetic cost to the plant to get carbon out of the air is less. And of course, the plant can use that carbon elsewhere for other growth processes. It doesn't have to use photosynthetic carbon to build these amino acids to build protein. It can use it for other growth processes. Okay, so the point is just to say that these organic forms of nitrogen are very efficient for plant growth. Key reason being is that you've got free carbon embedded in there already. Okay, and that's, again, the classic view of plant nutrition, of nitrogen nutrition is, well, we take whatever, it could be soil organic matter, but proteins in this example. What are we doing? We have to, the plants can't take them up. We have to break them down. So again, microbes will use those scissors, those enzymes and things to break proteins down into smaller components. Here's those peptides I was talking about. And then we break them down further into the amino acids. And then those are further broken down ultimately into the inorganic forms of nitrogen. And, the classic plant nutrition focuses heavily here. We say, oh, we want organic matter to cycle. We want it to mineralize. We want it to release the inorganic fractions, which then the plant takes up. Okay, that's the classic view. But as we've just pointed out, well, the plant can take up back here, the plant can take up aminos. It can even take up proteins, more complex molecules as well. So as these things break down and as the plant takes up the inorganic forms, what does the plant do with this? It doesn't want nitrate, as I said, it doesn't want ammonium, it wants amino acids. So it has to take up that inorganic form. And what does it do? It has to expend energy to rebuild, turn that into amino acid, and then again, turn that into peptides, and then again, turn it into proteins. So why do we have this kind of school of thought about the importance of these inorganic forms of nitrogen for nutrition? Why don't we, what's called metabolic shortcutting, why don't we just deliver the nutrients further along the chain? Why don't we just give these to the plant instead? Instead of trying to give it the inorganic stuff, let's just give it this. And then the plant doesn't have to expend energy on making all those enzymes to build this and then build this, and all this cost metabolic energy to build these proteins. Let's just deliver proteins directly into the plant. Metabolic shortcutting, save the plant's energy, it's more efficient. Okay, that's the key summary. Yeah.
You're in trouble. They gave me a mic. Just curious if you could elaborate a bit on some forms of amino acids in these proteins that we could, as producers, potentially use versus using the synthetic versions.
I sure can, yes. And I'll do that in a couple of slides away. Oh, where is that slide? Oh, maybe not. Did I take that slide out? Oh, whoops. So lucky you asked that question. I'm sure I had that in there. Okay, what am I talking about? We're talking about things like, I had a list here. For some reason, that slide is not there. Okay, so we're talking about things like, I think why protein hydrolysates are really interesting. Oh, sorry, it's in the next presentation. I knew the slide was in there. It's coming up in the next presentation, that's why. Anyway, I'll give you a short answer and we'll come back to this. But okay, so things like fish hydrolysates, protein hydrolysates. So taking fish frames and fish waste type materials and doing this enzyme kind of digestion and breaking them down into aminos and things. These are the types of inputs that I'm referring to, but you can make protein hydrolysates with all sorts of waste materials. It could be blood and bone meal type things, other industrial or agro wastes from pulps and pumice and residue. I mean, you can kind of take anything and enzymatically digest it and break it down. But, you know, so fish are a big one. But then other things like soybean meal or alfalfa meals, any of those kind of waste products or byproducts as well. Anything that's high, that you classically associate as high protein, if you kind of digest it and break it down, is the types of inputs that you could use to kind of make them yourself, or use kind of powdered meals, ground up and powdered meals to kind of provide some of these organic type sources. Now, anything that's high in protein, basically, is the answer. And I will come back to that in the next presentation too. Okay, so that's the answer. Let's just give the plant directly this complex stuff and save it all its energy on having to process it internally. But can plants take up protein? Can they take up these complex forms? Well, the answer is yes. Again, this is a really nice kind of summary chart. I really like this one. So yes, we can supply inorganic nitrogen. And yes, we have a very big understanding of the role of those in plant nutrition. They were the kind of the dominant model, the dominant thinking. So we have good understanding of how that works, how the plants take those up and how they're used. But it turns out that yes, the plant can take up amino acids directly and peptides and even big proteins. And we now know that the plants specifically release these enzymes called proteases, enzymes that liberate protein and amino acids from the organic matter. We used to think that plants did not do that it was only microbes that could do that, but we now know plants directly release these. So yes, they are trying to get protein and get nitrogen out of soil organic matter where they can directly take that up. And as we now know, many of you would have come across this rhizophagy cycle, this kind of rhizophagy concept. We actually know that plants will absorb whole bacteria directly into their roots. They will engulf and absorb bacteria into the roots and then use those bacteria as a source of nutrition. So if plant roots can take up bacteria who are much bigger than a protein, then yes, plants can take up complex proteins through their root systems. Those are a lot smaller than what a bacteria is. So if we can do bacteria, we can definitely do those. So, so this is just that point to say that yes, our understanding of these fractions in plant nutrition is minimized as we increase the substrate complexity. This is a simple ion. These are more complex molecules. As we increase the complex molecules, yes, we understand less about them from a plant nutrition point of view, but we are beginning to unpack that, and there's a lot of kind of work happening in this space. So yes, we can use organic forms of nitrogen like proteins, like protein hydrolysates, as a very efficient source of nitrogen for plant growth. Here's a study. I share this one just for interest. I thought it was really quite interesting on maize. We're looking at feeding the maize, looking at kind of root traits, how we can improve root traits, root biomass and whatnot. And we're looking at 3 different forms of nitrogen. We're comparing inorganic nitrogen, we're comparing amino acids, and we're comparing a protein hydrolysate. Now, all three of these treatments received the exact same concentration of actual nitrogen. So the content of nitrogen was the same across them all, just in the inorganic form or in the amino form or in this protein hydrolysate form. And the protein hydrolysate form is basically going to be kind of a mixture of, a protein hydrolysate is kind of a mixture of all of these organic fractions. So some of the more complex stuff as well as some of the aminos. And as you can see here, we're basically measuring a bunch of various root traits, root mass, root length, kind of lateral root production, all sorts of root various traits is what we're looking at here. And the black is the inorganic nitrogen, the synthetic nitrogen. The gray is the amino acid form, and the white bar is the protein hydrolysate, that more complex mixture. So you can see this general clear trend. Remember, the nitrogen is the same in concentration is the same in these, just the form of that nitrogen is different. And you can see here visually that, yes, aminos are more efficient than nitrogen, but the protein hydrolysates are even more efficient again. And we can see this in various root traits that they were kind of measuring, these, root lengths and lateral root production, all these kinds of things. So I thought that was very interesting in the way that clearly these protein hydrolysates were inducing these better kind of root systems, despite the fact that it was the same nitrogen content across the board. Okay, a little bit, we're almost getting close to the end. How am I doing on time? Okay, not too bad. A little late here, but I think I'm going to go a bit over, but I think that's okay. So just, I want to share a few things here about, again, some interesting quotes here from some papers that are like looking into this whole idea of organic nitrogen and its kind of role and function. I just want to share a few interesting quotes here from this one that was trying to integrate what they would call soil bioavailable nitrogen and how this whole kind of fits in. In agro-ecosystems, global nitrogen fertilizer use efficiency remains stubbornly low, around 40, 50% nitrogen use efficiencies typically. But of course, we need to double that and all of these predictions about the future and all of that kind of thing. I thought this was interesting, the modest success of technological focused solutions on fertilizer management reveals the shortcomings of this narrow focus, like your question on nitrogen inhibitors. And I mean, again, not to detract from them, I think they're an important piece of the puzzle, but we've had this very kind of focused view on tools and technologies to try and improve the use of fertilizers, improve the use of inorganic nitrogen. There's a broader conversation at hand. What about these organic fractions? What about some of the studies I was just showing you, highlighting that the organic fractions can be very beneficial? We haven't been having that broader dialogue. It's been very narrow focus on kind of techno solutions for nitrogen issues. And okay, their work calls for active management of soil organic nitrogen and suggests future agronomic research should seek ways to develop ways to enhance nitrogen supply from the particulate organic matter, from the mineral associated organic matter that we talked about. So actually, again, not be afraid of using soil organic matter. I'm going to kind of share an example here in a second. It's okay to deplete soil organic matter as long as you replenish it. It's got to be cycling. We want it to cycle. We don't want to burn it, but we want to also use it to help feed crops and also to keep it cycling. So that's what they're saying. We use it when it's high, but equally rebuild it during the off season or in the fallow seasons, this kind of thing. And so just pointing out, kind of bringing some of this together. Remember, when litter comes into the system, that plant residue, it starts to break down. It's in that various continuum, that state of decay, forming what's called that particulate organic matter. As that breaks down to small enough components, maybe some of those proteins or amino acids are released. So now we have an available, they're calling bioavailable nitrogen here. Of course, that can then be taken up directly by the plant, as we now know, could be taken up by the microbe. The microbe could be spitting out their waste products, cycling those nutrients. The plant's taking all of that up. As we discussed, all of these interlinking and getting stuck to those mineral particles, forming that mineral-associated organic matter, all of these kind of in this tangled web and interactions that we were kind of discussing earlier. Okay, So yeah, worth waiting for this slide. So this is going to just really kind of summarize some of this nuance really nicely. I'm just going to read this out off the slide here and then kind of just highlight dive into the picture here. So here then we're looking at the effects of nitrogen fertilization on that nitrogen supply on this mineral associated fraction. We're kind of specifically diving into this more nitrogen rich, that more nutrient rich fraction, that mineral associated one that we talked about earlier. So on the left-hand side, modest and economical fertilizer application. Okay, again, it's not to say none, but it's just saying, you know, small amounts, targeted amounts, just not overdoing it is always the key. So modest economic applications incentivizes the plant to invest in root production. Okay, we talked about this in detail. We are incentivizing the plant to invest in root production and those associations with mycorrhizal fungi when we feed them modestly. When there's light nutrition available, the plants are going to go searching for it. They're going to expand their root biomass. They're going to look for those nutrients. They're going to form those associations with the mycorrhiza so that the mycorrhizal hyphae will also go further beyond and explore greater soil volume, trying to look for those nutrients and that moisture, etc. So this is the key point. You know, and I mentioned those organic forms of nitrogen. They are also processed down in the roots. They encourage this greater root development. Okay, so this is this key point. And do you remember I showed that study that also showed that, again, nitrogen fertilizers negatively affects microbial biomass carbon, but also mycorrhiza. That was the other big one that it also had a negative effect on. And again, I talked on that the fungal necromass was a more important contributor also to that stable soil organic matter. Okay, so we are forcing the plant to invest more heavily into below ground, into roots, into root production, mycorrhizal associations in pink here. So the resulting plant microbe mineral interactions in these modest fertilized soils. One, this liberates more bioavailable N from the minerally associated organic matter. If the plant is a little hungry, if it's light on nitrogen, yes, it will mine the nitrogen from that soil organic matter. It will liberate some of that nitrogen from that minerally associated nitrogen-rich organic matter. It will do that. It's not necessarily a bad thing. It will liberate that nitrogen. Two, it will increase then microbial biomass. The microbes, if they're hungry, they will go looking for that nitrogen. They will liberate that nitrogen. And that is a very form that then they can help. It helps them get the carbon, get the nitrogen, grow their microbial biomass. So we're encouraging the microbes to increase their microbial biomass. They will also produce, this system will produce less wastes, less nitrogen losses. The more inorganic nitrogen we have, it's the very reactive form, it's very leachable, it's very volatilizable, it's very easily lost from the system. So less inorganic means less losses there, and ultimately increasing necromass inputs that can then replenish those minerally associated pools. So this is this point about drawing from and then replenishing. So to summarize, in these modestly fertilized soils, just a little bit of green here, okay, the bacteria, they're hungry, they go looking for nitrogen. Yes, they will mine it, but we liberate lots of nutrients from that organic matter that encourages more microbial biomass production. And when those microbes also grow and feed off that and grow more microbial biomass, okay, then they end up dying, and then they form more of that microbial necromass and help to replenish that supply in which they took from. Okay, so it's just, in summary, it's giving them a form of nutrients that's very conducive to cycling for them to grow and then to die and replenish and put back. But when we over-fertilize, we are basically interrupting those cycles. We're not, the heavily fertilized systems, we're not forcing the roots to grow, we're not forcing those mycorrhizal associations. This is often argued as a good thing because it doesn't induce that nitrogen mining from the soil organic matter. It's this idea that we should fertilize because then the microbes won't go hungry, then they won't mine that soil organic matter, the nitrogen from that soil, minerally associated soil organic matter. That's the argument. No, let's feed crops, let's preserve our soil organic matter. That's the argument. And in many instances, that could well be the right thing to do. But when we bring in this concept of the organic nitrogen and how it's embedded in the organic matter, it's just a bit more of a nuanced kind of dynamic here, suggesting that Okay, when we do that, yes, we preserve that. However, that doesn't lead to this then cycling or the replenishment that what we see in these more modestly fertilized systems. So it's a nuanced picture. I'm sure there's a whole range of different contexts and arguments as to when it's this way or that way, or what we should or shouldn't do and whatnot. But bringing in this idea of organic nitrogen, this is kind of some of the latest thinking. This is very recent study here from last year, highlighting that when we think about this quality of this soil organic matter and its contribution for fertility cycling, but also to be more supportive, it's a form of nitrogen that's more supportive for microbes to help them grow more microbial biomass, which ultimately, again, increases that necromass. So again, thinking differently about nitrogen use efficiencies. Actually, this is a paper from the University of Saskatchewan. I'll just read a few of these quotes. Nitrogen use efficiencies for today and tomorrow. For a more complete picture of measuring nitrogen use efficiencies, the entire plant should be considered, including the roots. Roots comprise roughly 14% of the total plant nitrogen in annual crops, with 4 to 71% of the total plant then released back as rhizodeposits, so through root exudates, through cycling, et cetera, et cetera. A conspicuous oversight in how several nitrogen use efficiency indices are conceptualized is to neglect and not consider the organic forms of nitrogen, only focusing on inorganic, focusing what we apply in the inorganic form and how much of that we're capturing, and having those discussions about nitrogen use efficiency purely from an inorganic nitrogen point of view. To better capture all of the forms of nitrogen that plants use, the reconceptualizing of nitrogen use efficiencies must include soil organic nitrogen pool that is available to and usable by plants, as this would numerically reduce several traditional estimates of nitrogen use efficiencies. So really what we're saying here is that if we did broaden the concept of nitrogen management and consider the organic forms, actually many of our, what we consider our nitrogen use efficiencies would get worse, would be lower, when we start to factor in these organic pools. When we only narrow it down and think about the inorganic pools, it looks better than it kind of really is. So I think this is a really good summary slide as well. This kind of really captures all of this kind of detail that we've really dived into for the past few hours, and really just summarizes it really nicely. We're talking about inorganic nitrogen here versus organic nitrogen here. And I just kind of, again, it's a balance, the representation of the scales here. You're always going to have a little bit of both. It is both, but it's just really about how the balance is tipped. Is it too much one way versus the other? It's about having a little bit of both. When we have more inorganic nitrogen forms cycling, this encourages shoot growth, as we mentioned. These systems are more leaky. A lot of inorganic nitrogen is very reactive. It's going to leach, it's going to volatilize. So we see more nitrogen losses, very leaky systems. Encourages more shoots, not so much roots. And generally, if the scales are tipped too heavily in this way, this is what's going to lean more towards soil organic matter decomposition. This is where we're going to be more so perhaps burning the soil organic matter. However, if we lean towards the organic nitrogen sources, we have more root growth, as we touched on, more stronger association with those microbes, a bit more of a carbon surplus, free embedded carbon, as we touched on as well. So when we lean more towards organic forms, generally this is going to take us in a little bit more of a soil organic matter building, soil organic matter stabilization direction. I think this is just a really nice kind of summary of that. It's going to be a bit of both. It's always just about that kind of a balance, but as a generalization, the more we can steer it towards thinking organically, organic nitrogen, you're going to be more towards building soil organic matter. So what are some of the organic sources of nitrogen we can use practically? Let's get a few last slides on practical. Let's take a quick question first. This is a good time.
Where do foliar applied nutrients fit into this whole discussion?
Yeah, great question. They fit in a lot, and it's not the topic of discussion for today, because it's a big one. No, I'm a big fan of foliars for this very reason, which is that by providing foliar nitrogen, you can really keep a production focus on. You're delivering that for the plant. It's not really for the soil. It may be an inorganic form of nitrogen, but when it's going into the foliar, that's quite okay. It's not going down into the soil. That's the beauty of the foliar approach. You're not putting so much of these very reactive, leaky nutrients down into the soil. You're just delivering them straight into the crop. where the plant can take them up and then turn them into organic forms within the plant tissues. So yes, I'm a big fan. Wasn't the topic of conversation for today, but if any of you heard me spoke before, you know very well that I'm a big fan of the foliar approach, and we could have a huge long conversation about that too, maybe next time. So yes, I think it's a really good idea though to use nitrogen through that pathway, keep the production focus on, but you're kind of keeping it out of the soil and optimizing some of these interactions.
Okay, I've got two more.
Sure.
Do traditional soil tests measure organic and inorganic nitrogen levels accurately?
Ooh, that's a good question. So one, I would say a traditional soil test typically doesn't even measure it. We often will only be looking at the inorganic forms. If you've got a measure of your soil organic matter, then you could get a bit of a rough calculation out of that as to how much kind of organic nitrogen may be embedded within that soil organic matter. So it's kind of more of a calculation fudge factor, but it would give you a bit of an indicator. It's not commonly done. It's very laborious and very arduous to kind of measure, so to measure all those amino acids or to be measuring some of those proteins. It's a very kind of complex picture. So it's not normally done. It's normally kind of more so guesstimated from the soil organic matter. That's probably the best answer. So no, we don't normally test for it, but maybe obviously we need to work out ways to do that more effectively and more efficiently and cost effectively, etc.
Okay, and do you have a perspective on the degree to which modern standard soil tests are able to make recommendations for inorganic N and P that optimize the use of minerally associated sources in annual cropping systems?
Yeah, sure. So that's a good question. That's the real challenge. That really comes down to that challenge that I was talking about, you know, of really optimizing nitrogen inputs for optimizing root growth. You know, too little and too much induces problems. So it's really about trying to optimize that N application rate accordingly. I would kind of answer it a little bit in the same way. That's a very difficult question to answer because as we pointed out in this last slide, it's not to say that, you know, it has to be no nitrogen, you know, no applications. It's not that it has to be that. They're pointing out that it's about modest applications, again, kind of in that sweet spot, in the Goldilocks kind of zone, where, okay, small amounts are quite okay. You need small amounts. That's going to help biomass production and et cetera, early on. Just not too much that then begins to shortcut some of those associations with the microbiota, et cetera. So the answer is kind of the same to that. It's just about that sweet spot in order to optimize the cycling, but how to predict that from a standard soil test, I would say no. I would say we don't really have that information yet. There's so much research happening on all this whole stuff we've talked about today, this idea of particulate and minerally associated organic matter. It's a big field of soil science and soil carbon science at the moment. There's a lot of papers, a lot of work happening in this space. So I'll admit there's a lot of unknowns. We have a lot of blind spots in this discussion. I'm sharing it as the best as I can in this kind of level of detail that we'll try and bring it to some practical applications now. It is a complex picture and we do have a lot of knowledge gaps. So I would say no, we don't really probably have very good ways to predict from a standard soil test where that optimum rate is to ultimately kind of preserve and optimize this kind of cycling process. And that would be my best way to answer that one, a bit long-winded and roundabout. Okay, so then just to wrap up, and I'm sure you're all desperate for feeling a bit overloaded and desperate for a coffee. So what are we talking about here in terms of what's going to be the easiest way to get organic nitrogen into the system? Okay, what are the options here? Okay, of course, it's anything, any of your crop residues, those roots, those root exudates, the microbial waste products, microbial metabolites, all of these kinds of things, those dead organisms that we also touched on, the microbes, other insects and their wastes and feces, etc. And of course, organic amendments. I mean, it could be applying composts, applying manures. You know, yes, you're going to have some organic forms of nitrogen coming in through those. And again, lots of good studies that show actually, again, an integrated approach, you know, using some compost and manures with synthetic nitrogen. This is a very good tool, a good strategy to build soil organic matter. Plenty of studies show that a combination of inorganic and organic forms is a very good kind of pathway forward. So, okay, if you can use compost or manures, excellent. But I know that's on broad scale. Sometimes that becomes very difficult. So the obvious answer is legumes, bringing legumes into the picture. They're going to get some free nitrogen out of the air. They're going to deposit that nitrogen into the system in organic forms, be that through, again, their root exudates from living roots. The legumes particularly, they're going to release, they exude a lot of amino acids. Their root exudates are very nitrogen rich. So we get amino acids from legumes in real time from living roots. But of course, as the legumes die and decay and their residues, of course, that's a lot of organic nitrogen coming into the system through the nodule decay, through the root decay, through the shoot decay, et cetera, et cetera. So one of the good things then, I really think this idea of bringing legumes into the picture, especially in a companion cropping, intercropping type scenario, is that we can also help to prevent this issue of, nitrogen mining. If we've got a lot of, I know it's the right thing to do, our high stubble farming systems in dry areas, the same here, and in Australia it's the same deal. Yes, we want lots of high stubble, moisture protection, residue, all of sun protection, all of those benefits for dryland systems. Yes, we want these high stubble farming systems, but again, they're also an overload of carbon. We can still get nitrogen organic matter. We can still get soil organic matter mining for nitrogen. It's not just about mining for carbon, it can also be mining for nitrogen. I showed that slide earlier. So simply by including some legumes in the picture, we're bringing a bit of highly available nitrogen into the system in an organic form, whereby the microbes can then use that in combination with all of the benefits of stubble and protection, et cetera, plus provide a little bit of available nitrogen to prevent nitrogen mining from the soil organic matter. So I think legumes, I know they have a bit of a bad rap in some circles and in some thinking, but I think that they could be a really useful piece of this puzzle to optimize this process. Okay, it doesn't have to be a legume cover crop or an off-season thing either. We can do it while we're cropping. That's the beauty of the intercropping system. A nice picture here of some canola and peas. I mean, it could be any other combination, of course, but a legume cereal combination, I think, is a really good one. There's your classic kind of piola, of course. And again, another one, just to show a nice visual image. And you guys would be laughing a little at this picture. One, maybe the size of the combine, but to look so lush in the understory during harvest time. I know it's so much dry here at harvest. This is from the UK, but I just visually it illustrates just a really nice example here of, litter, residue, high carbon, but in the understory. Okay, he's obviously got enough moisture there to kind of have a really nice green understory with some legume, with some clover there. We've got a perfect combination of the two different litter types, nitrogen-rich litter, highly labile, highly available, highly digestible from the microbes. They can rapidly convert this into microbial biomass and then necromass and then that minerally associated organic matter. But okay, we also still have the stubble and the residue that's going to give us the protection. The poorer quality litter, it's still okay. It's going to break down a bit more slowly and form that particulate organic matter that we also talked about. So yes, so this was just to illustrate that, that legumes, they can transfer nitrogen in real time during the growing season from living roots. It's not just about legume residues and litter decomposition for next year. We know that legumes will release root exudates, amino acids, peptides. Again, they are also released as root exudates. So those amino acids that are excreted, again, if we have root intermingling, here we have a non-legume, a nitrogen scavenger, a cereal, it could be an oilseed, whatever, it has a high nitrogen demand. As soon as those roots are intermingling, the legume is releasing those amino acids as root exudates. The cereal or the grass, whatever, it's going to be scavenging those. It's going to be taking those up. It has a high nitrogen demand. So we will get transfer through root exudates, but additionally, if both plants are mycorrhizal, we also have mycorrhizal fungi connecting the two plants. We would also see, again, amino acids transferred directly to the non-legume through the mycorrhizal fungi. So in the current season, in real time, there's all these kind of nitrogen sharing kind of dynamics at play. So okay, how do we integrate this into some like strategies that we can kind of use, practically speaking, and we can kind of elaborate this in some question time, some discussion. We'll probably go straight for a quick coffee break and maybe come back and have some discussion. I'm sure you're all a bit itching for some coffee. So, it just kind of summarized some broader points or strategies that we could kind of be thinking about and implementing to kind of help all of this process that we discussed today. Carbon-based inputs. So again, organic forms of nitrogen, like we were saying, but I also define carbon-based inputs as this idea of putting carbon with fertilizers. So putting things like humic acids or fulvic acids with nitrogen, using seaweed extracts or amino acid, protein hydrolysates with nitrogen. We're going to talk about this in the next presentation, so we'll get more onto this, but this idea of carbon-based inputs. Anytime you're putting fertility down, include a carbon source with it. As we just touched on there, intercropping with legumes, I think that's going to be a really useful piece of the puzzle. Sure, if you can, use organic amendments, compost manures wherever possible. Could also be livestock integration. Why not? You're going to get the manure component from them. That's going to be really advantageous to provide organic nitrogen through that manure. So I think wherever possible, livestock integration. Foliar nitrogens. Yeah, I was thinking ahead for the questioner at home. Yes, foliar nitrogen is great. You're putting that into the crop, you're keeping it out of the soil. I think that's a really good strategy, hence why it's there. Again, maybe combining other synergistic nutrients with that or with nitrogen inputs. Nitrogen doesn't work on its own. This is another topic to go through. Nitrogen nutrition wasn't for today, but nitrogen doesn't work on its own. It needs sulfur to help build proteins. It needs molybdenum to convert those nitrates. We could go on and on. It needs magnesium to form chlorophyll. There are lots of other nutrients that work with nitrogen, help it utilize and be metabolized in the plant. So it's not just about managing nitrogen itself. It's also about the other supporting nutrients that work with that nitrogen. We could be using biofertilizers. Why not some N-fixing bacteria? I'll talk briefly on this in the next presentation. Let's bring some free nitrogen in through some of these N-fixes. It's not just legumes and rhizobia that fix nitrogen. There are many other free-living nitrogen-fixing bacteria who associate with cereals and legume and other non-legume plants. And sure, things like longer-term game, plant breeding, optimizing nutrient use efficiencies through plant breeding, bigger root systems, more of that nutrient scavenging, more of that microbial associating, all of those kinds of things that we also kind of talked about. So in summary, soil, organic carbon, and nitrogen dynamics are complex. There are many knowledge gaps and a lot of rethinking, new ideas emerging. Some of the stuff I've shared with you today is very current new thinking. A lot of those studies were from just last year or the last 1 to 2 years. Some of this stuff is very current. There's a lot of rethinking happening. It's going to be your job, obviously, to translate this into practical applications. And okay, we can have some question time in after the break, perhaps on that. But just to let you know, this is kind of where all of this is going. But there are many knowledge gaps. But the emerging paradigm on soil carbon, it's challenging some of the old ideas, this contribution of microbial necromass. If we acknowledge that this makes a big contribution, it changes the discussion on, well, how best to build soil organic matter. Maybe root exudates are much more important than we traditionally thought, because those exudates feed biology so efficiently. We can grow more of that microbial biomass, which can then form that necromass. So again, we're challenging some of the old ideas, and it's a very interesting time. Nitrogen inputs. I know it's not an easy answer, but they should be optimized as best as possible. Excess nitrogen and those negative effects on root biomass production, those negative effects on microbial biomass that we touched on. So trying to optimize that as best we can. Carbon-based inputs, again, so that's what I was talking about just there, to kind of create an artificial organic nitrogen. So just combine humic acids or carbon things with the synthetic nitrogen, you get this carbon-nitrogen bond, makes the nitrogen behave a little bit more like an organic nitrogen. It's got carbon embedded in it. We talked about that embedded carbon with the amino acids and how that helps with efficiencies, etc. So yeah, use organic amendments where possible, but otherwise I think intercropping with legumes and this legume piece of the puzzle has got to be a really easier way to kind of practically translate some of this. So all of those discussions that then come with how to intercrop and how to optimum seeding rates and blah, 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 and all of those other things is great to see all of that work happening at the moment and so much traction in the intercropping space. I think that's really good. Okay, we got there. Well done to you all. You all made it.
Thank you very much, Joel. That was awesome. We're going to take a 15-minute break. We're going to try to get a little bit closer to on time. Anybody that needs to use washroom, they're at just outside the doors at either end of the hallway. We'll meet back here at 3:40 for part 2.
Sounds good. Questions on that session? Yep.
Yeah, hi. I'm not sure if I'm pronouncing it right, but that ability for plants to suck up peptides and proteins and amino acids, was that element called proteases? Or what was that?
Unless you're thinking of protease.
Protease, yeah. So protease, is there anything else that enable the plant to utilize proteins, peptides, and amino acids directly?
Yeah, so that's what the protease is, the enzyme that the plants release, which then goes to the organic matter and breaks it down and pulls out the aminos and the proteins and liberates them so that they can be then taken up. So it really, otherwise, apart from that kind of direct process, it would be really just trying to encourage microbial activity, because those microbes can also release all sorts of different protease type enzymes that can also liberate that. So it would be kind of, the general answer would be trying to encourage more microbial activity, and then they will help to make a contribution to that as well.
Okay, just one follow-up there. Are there plants, you're just referring to plants in general, but are there, like, are annuals or perennials exude more of that, or within annuals, cereals, pulses, oil seeds, anything like that would have a different capacity?
Good question. I don't know. Yeah, there probably is some plants would do it better than others. That is very likely, but I don't know. Yeah, I've not come across that, so can't help you on that one. Sorry. Yeah.
Hi there. You had a slide showing the relationship between nitrogen fertilizer and our root size. And I was also wondering if you had a comment on the relationship between plant root exudates and nitrogen fertilizer.
Sure. I actually had a slide on that and I skipped over it just to save a bit of time. I was going to show a slide that showed it was, however, it was a hydroponic study, so nutrient culture. So how much ground truthing we can kind of extrapolate from that is hard to say, but it was in a hydroponic study that because it's much easier. Okay, so measuring root exudates is very difficult, and it's very difficult to do in soil in situ without disturbing and getting them, etc. So a lot of what we know about root exudates comes from hydroponic type studies, where then the plants, of course, are exuding those exudates into the solution, where it's easier for us to then harvest that solution and measure the presence of the exudates in there, in that water, in that growing liquid. So, so there's difficulties there to then extrapolate that to field conditions. But I was going to share a slide that showed that, yes, that they were looking at the impact of nitrogen on root exudation, and showed that as you had none and very low amounts and increased the nitrogen rate, showed that you got increases in root exudation. So they did show an increase in nitrogen led to increase in root exudates. But of course, it only went up to a certain point. If they kept going higher on the nitrogen level, it would have probably started to also decline. So I am making an assumption that, again, it's going to be an optimum, because they showed a really nice dose relationship where a little bit helped and a little bit more nitrogen helped more root exudates, a little bit more helped more. So it was that kind of dose relationship. And we could assume that as you start to go higher on nitrogen, like any nutrient, it starts to become potentially toxic, or, you know, interferes metabolically, counteracting other nutrients, and then that's going to induce other limitations and therefore potentially likely reductions in root exudates as well. So I've only seen that one little hydroponic study that I could draw from there, but yeah, my guess is it'd be a similar relationship. Yeah. Okay. Good. Okay, so if I could have my slides, please. I promised you guys a little less science on this one. There's still going to be some. There are still going to be some. I'm going to share some studies and things, but we'll try and keep this one a little bit lighter and a little bit more free-flowing, hopefully. So how was everybody with that last presentation, though? Was it pitched at an okay level? Were you with me? I'm sure I might have lost some of you bits along the way, but was it for overall, was it okay? Everyone kind of got the key point there? I hope so. That's okay, good. It's a bit of a complex one, obviously, as you saw. Yes. Question from online.
Yeah, we have a question from online. Can you please comment on a symbiotic free-living nitrogen fixation? Kula bio
Yes.
Ustria?
Yes, okay. I also was going to talk a little bit more on that, but I did kind of I mentioned it just only briefly in the presentation. I was going to expand a little bit more, but I decided just to, we needed to wrap up there, so I didn't go deep into that. But just to point out, and I did touch on this, but just to point out that yes, it is not just rhizobia, it's not just legumes that can fix nitrogen. There are many other what's called free-living nitrogen-fixing bacteria that live in soils that can associate with any crop, any plant, a broad range of plants. These are often called kind of associative nitrogen fixers. So there's lots and lots of studies showing that there's plenty that associate with wheat or with maize or with all of the cereals, et cetera, et cetera. So yes, it's not just to say that legumes are the only ones that can bring nitrogen into the system and contribute to that. Indeed, even cereals can, with these other free-living nitrogen-fixing bacteria that, okay, they're not rhizobia, they don't form nodules, but they just live around the root system and feed off the exudates and return nitrogen. There's many endophytes, nitrogen-fixing bacteria that live inside the plant, up inside the stems or within the leaves, and can also fix nitrogen from inside the plant. There's a few products that have been commercialized for this too also. So yes, it's a good question. And yes, there is a lot of opportunity there to use some of these free-living associative end fixes with non-legume crops. And it's an obvious thing that we should also be doing. It's going to help be a piece of the puzzle, help use a little bit less of the synthetic nitrogen, but also help us provide free nitrogen, but also free nitrogen in an organic form for directly into the plant as well through those bacteria. So for sure, there's opportunities there to use those types of inputs. And I see there's more and more of those products kind of being commercialized. And sure, like all biological products, I guess we'll talk about this quickly now. Well, it'll come up in the topic, but of course, there's a lot of interest in the role of microbes and microbials and these kind of biofertilizers. And rightly so, I think that's good. But of course, there are hurdles. Some of these products, they can be a little inconsistent. They can work very well in the lab and not always so well out in the field. So there are some hurdles. We do have some fine tuning to do in order to optimize the efficacy and functionality of these kind of microbes out in field conditions. But clearly the evidence is there, the potential is there. Sure, it just maybe needs a little bit of fine tuning and ground truthing, etc. So, okay, so the topic for this afternoon, leveraging soil biology for nutrient cycling. How do we use biological processes, biological organisms, by that I mean plants and microbes, to unlock nutrients in the soil, to access nutrients in the soil, to scavenge, to access and release those and cycle those and make those available. That's going to be the theme for this afternoon. So we've got kind of 4 main parts to this talk. We're going to do a bit of an introduction, just kind of set the stage, and then really talk about these 3 tools, 3 strategies, using plants to scavenge nutrients, using biostimulants or kind of feeding the organisms, so stimulating them or giving the microbes food sources to activate them, or inoculating them, so using things like biofertilizers, actually putting organisms into the soil. So these are kind of the pieces of the puzzle that we will kind of discuss. So, and just kind of then set the stage with this introduction. what is the potential? What are we talking about here? Let's kind of start by thinking about our soil nutrient levels. And many of you would have heard me talk about this kind of thing many times before, but just to start by pointing out that when we do a soil test and when we look at soil tests and look at the nutrient content on them, often we're just measuring the more available pools, what might be considered more plant available. And it's not that we're measuring everything that's there, it's just the fraction that is more available to the plant. Which is important, that's part of the equation, but it's not the whole story. There is more nutrients in soils that are present in soils, but they're not in available forms. They're kind of locked up, bound up within soil organic matter, for example, or mineral associations, like nutrient to nutrient kind of tie-ups and these kinds of things. So there's lots of nutrients there, they're present, but they're just locked up or in unavailable forms. And this is the argument of trying to use things like biostimulants or biofertilizers, or use plants and cover crops and roots and their root exudates, etc., is to try and unlock and access those nutrients. So what we typically measure on a soil test, it's actually a very small part of the equation. What's soluble is a very small fraction. That's kind of what's in the soil water kind of thing, the nutrients that are floating around in the solution. And yes, they can be taken up directly into the plant from the soil solution, from the soil water. But then we also have other fractions of nutrients that are what's called exchangeable. This is a slightly bigger pool. These are also relatively available. These are the nutrients here that are kind of stuck on the soil surfaces, being held by the soil colloids, by the soil surfaces. And again, lighter sandy soils, of course, have less surfaces to hold organic matter like we just talked about. Similarly, sandy soils have less surfaces to hold onto water or nutrients equally in the same way. So versus a clay soil that has much more of these colloids or charged surfaces to hold on to nutrients and water. So these are the what's called the exchangeable nutrients. They are sitting there stuck to the soil surface, soil particles, and they're called exchangeable because the plant can exchange them. The plant can release those root exudates, release those acids, release hydrogen, where those will displace potassium in this example, displace the nutrient and exchange it, and so that the plant can then take it up. So these are the two pools that are both relatively available. You're soluble and these exchangeable, they're fairly available. But they're a small fraction of the total pool that exists in the soil, and embedded in the soil is other, again, nutrients that are kind of bound up, locked up with each other, or bound up within the clays, or the kind of the parent material, maybe not fully weathered yet, these kinds of things. And just so then to point out that is the reserve that we want to tap into. This is the goal of using biology and biological processes to access that. And just to point out that it is significant. These numbers, the amount of nutrients that are in your soil can be significant, tenfold more than what you see on a soil test. There's much, much more there. It's just that it's not available, just not in a soluble or a highly available form. It's in more of these unavailable forms. So let's have a look at just some examples. I'll just, again, just quickly point this out. We won't dive deep into this. I'm just going to point out, here's a couple of the macronutrients here. And we're just comparing what a soil test that uses a weaker extractant that gives us a picture of what's more available versus using a soil test that uses a very strong extractant, strong acid that strips everything off, gives us more of a picture of the total pool of nutrients. So just as an example to kind of show you this general trend, the general relationship, here's a fraction of calcium that's available, but the total pool is, 5,000 versus 38,000. There's a lot there. We could go on and on. It's the same trend, 400 versus 14,000 for magnesium. Sulfur, 7 that's available, yet 200 odd in the total pool. Phos would be, of course, an important one. We have low phos soils here out west. Phos is a big problem. Actually, this is from a Saskatchewan soil. The total pool, there's 400 odd parts per million of total phos. It's there, it's just kind of locked up and bound up Okay, in a very alkaline soil, that phos would likely be bound with a lot of calcium, forming a calcium phosphate, an insoluble, unavailable calcium phosphate that is there, it's present, but it's just not highly available for immediate uptake. The plant has to work at that. It's got to release very specialized acids and enzymes that can break apart that calcium phosphate, liberate the calcium and the phosphorus, and take those nutrients up. So again, just highlighting this is, you know, there's orders of magnitude more there than what's necessarily available in the test. Similarly, on some trace minerals, I mean, zinc would be another common limitation out here. I mean, half a part per million that's available, but there's 40 odd in the total pool. Copper and boron would be other examples. Okay, 1.6 available, 13 in the total. Boron, 0.3 available, 8.5 in the total. Okay, we're not going to go through all of these, just illustrating to you that there is potential there to be leveraged. You do have reserves in your soil. Now, if you have a very light, sandy soil, of course, your potential total pool is going to be much smaller than if you have a medium or heavy textured soil. Of course, clay soils are going to have way more total nutrients than a sandy soil. So yes, in a lighter sandy soil, your opportunity or your potential to leverage all of that fertility is less. You know, you've got less potential to do this because your sandy soil just simply doesn't have huge amounts of total nutrients. But medium or clay soils certainly do. So there's a lot of potential there. And with fertilizer prices the way they are, there's good economic incentive to use what you have there to try and leverage that, use some of these strategies that we'll talk about. So how then do we solubilize those nutrients? How can the plant access those? I touched on that just before, releasing organic acids or hydrogen to exchange nutrients, but also very specialized enzymes. These are compounds that can liberate that calcium and phosphorus, for example, or liberate the phosphorus from the soil organic matter. These are specialist kind of tools that the plant can release or the microbes can release that help to unlock and break apart those tight bonds or liberate and release the nutrients, etc. So enzymes or all sorts of various specialized root exudates, and I'll give some examples of these as we go through. We talked on this in detail just in the last talk. Sure, breaking down organic matter or cycling that organic matter. When we cycle that organic matter, we are liberating the other nutrients embedded. Organic matter is not just carbon, it's also got that nitrogen, phos and sulfur and everything else, little bits of calcium, potassium and some trace minerals, it's all in there. And some of those nutrients get liberated as we cycle and break down that organic matter. And then lastly, plant nutrients are made available when microbes die. Those microbes are feeding on nutrients from the soil, taking them into their bodies. And when those microbes die and their bodies break down, we had that question about what happens to necromass if it doesn't get stuck to mineral surfaces. You know, that necromass will eventually get decomposed and break down, and all of the nutrients inside the microbial bodies will also be released. Or when that microbe is consumed by a high level predator, when a bacteria is eaten by a protozoa, for example, that protozoa will eat it and process and metabolize and excrete waste products. And again, those microbial waste products are sources of nutrients that are getting cycled that then the plant can take up. So, of course, therefore, that really means at the end of the day, it's all about fostering that workforce in the soil, those microorganisms, feeding them, nurturing them, looking after them. It all comes down to photosynthesis. It's all about plants photosynthesizing, bringing carbon in, depositing that carbon into the soil through roots and shoots and exudates, depositing that carbon as food. And that's the food, the fuel to then feed all of the organisms, the bacteria and the fungi. They start that process, they degrade it, and they decompose, incorporate the nutrients into their bodies. And then all of these higher level predators, they run around eating each other. And as they eat each other, they're again feeding, metabolizing, releasing waste products like we all do, and again cycling nutrients for the plant uptake. So it's really all about, if we want to leverage fertility in the soil, we've just got to nurture the living component of soils that help to cycle that, to help unlock and cycle and liberate all of those nutrients that we have in the soil. And of course, in doing that, we can help lower our dependency on the fertilizer inputs, on those external inputs. That's the opportunity there that we have by fostering these microbial relationships. And as we touched on, when we use Too much fertilizers, yes, it can compromise these associations. We talked about that of the nitrogen and that dynamic of suppressing microbial biomass or suppressing mycorrhizal associations. We can have some of those negative effects on that association. So there's good incentive there to lower our dependency. And of course, the other big factor is that our synthetic fertilizers are generally not so efficient. We don't see great nutrient use efficiencies with fertilizers. Okay, nitrogen is quite an extreme example, 50-odd%. I mean, it depends on different soil types and whatnot, and different climatic conditions, but 40 to 50 to 60% at best. Nitrogen use efficiencies, 50-odd% of what we apply actually gets into the crop. So there's economic incentives there to improve these efficiencies. And key to that is not only the fact that then those nutrients are very leaky, particularly nitrogen, leaking from the system, causing environmental damage. It's bad for the back pocket. You can be saving money by using less fertilizer, but it also is very leaky. In the example of nitrogen, here we see nitrate leaching zones of the world. And you can see that we have a lot of parts of the world. There's a lot of nitrates leaching into the groundwaters, into the subsoils, etc. Particularly us through here, but many parts of Western Europe and many parts of Asia. Lots of nitrogen loss of the system, and it's a burden on the ecosystem, burden on the environment. Again, what effect does that also have on the biology, those organisms that we want to nurture and support? Here we have a study from the UK, from Rothamsted, long-term, over 150 years, looking at the effects of long-term fertilization, 100 plus years, on microbes. And as they find that plants and soil microbes have a consistent response to long-term nitrogen fertilization, both richness and diversity of plants and soil microbes are significantly decreased. And the key gene, the microbial genes that help to function and govern carbon, nitrogen, and phosphorus cycling, they are all suppressed. So our results indicate that century-long fertilization weakens this plant microbial network. So over long periods of time, we see less microbial activity, key genes being expressed that help to cycle these various important nutrients. And that also, not just at a microbial level, has an effect on the plant species diversity. This is from a grassland study. Here's the control with no fertilization. You can see much greater species diversity of grasses, of plants in the grassland, in the pasture. Under the fertilized system, we have narrowed the plant species diversity, less diversity present under this longer fertilization regime. So the negative effects, as we kind of alluded to and talked a little bit about earlier. And of course, the other obvious example is start of phos. We all know start of phosphorus is really important for crop establishment, and of course it is. But that can come at a cost where if we are supplying too much available phos, we're not going to form these mycorrhizal associations. The plant will not form these mycorrhizal associations quite so well because there's enough phosphorus there from the start of phos. So the plant gets a bit lazy. I don't need to form these associations with mycorrhiza. I've got start of phos there. And the trick, I know that we've got cold soils here, and there's a challenge there to get the crop established in such a short growing season. You've got to get things up and going. I know that's a real challenge here, of course, as well. But mycorrhiza, they don't just supply phos. They do lots of other things. They're really good at improving drought resistance, tolerance to salinity, disease resistance, tolerance to heavy metals. I mean, all sorts of other benefits that mycorrhizal fungi can bring. And we are missing out on those other benefits. Yes, we are supplying starter phos. And by doing that, we are short-circuiting that association and potentially missing out on all of these other types of benefits. It's not just about the phos supply. So these are important considerations, and when we're trying to foster and nurture these microbial associations. And just visually, again, you can see how important mycorrhiza are at helping the plant access greater soil volume. I mean, it's anywhere from 10 up to 1,000% increase in soil volume that the mycorrhiza can help the plant access. You can see there's your tap root, and there's the kind of the core of the plant root system roughly around this zone. And from there, the mycorrhiza have associated with that root system and grow out in all directions, exploring. Just visually, you can see visually this greater soil volume in which we are exploring, accessing moisture, accessing nutrients. think about this picture and the greater access to soil volume and superimpose my starting image with the circle of the soluble, exchangeable, and total pool of nutrients. So thinking about that big total pool of nutrients that exists, it's there but locked up, plus you're accessing greater volume of soil to then access that greater volume of that total pool of nutrients, especially at depth as well. There's big reserves of nutrients in soils. There is big potential to just use that fertility through these biological associations. Again, less in a sandy soil, more so in medium soils, but nonetheless. So, okay, what are some general strategies to improve nutrient use efficiencies here? Of course, things like integrated nutrient management, and that really just means use all the tools that you can. You don't have to manage fertility just with fertilizer from the bag. Yes, use livestock. Yes, use compost. Use manure. Yes, use some biofertilizers. Yes, use legumes. Yes, use an integrated approach of lots of different tools to manage your fertility. That's always got to be the approach. Same as integrated pest management. Same philosophy. So things like, of course, the 4 R's. This becomes really important. Again, organic amendments, carbon-based inputs that we kind of touched on. Things like seed treatments and foliar sprays, things I talk about a lot, but not necessarily the topic for today. But, we're still using inputs here, but we're using very small amounts, more targeted, not larger amounts broadcast into the soil. We're being very specific, very targeted with very small amounts right around the seed, or very small amounts being foliar sprayed directly onto the canopy. These are high efficiency mechanisms, high efficiency pathways to feed plants. And then, okay, the 3 we're going to talk about today. We're going to talk about using plants, using biostimulants, and using biofertilizers. And I think just to stress, I have all of these conversations about this. We're going to talk about some of this nuance of plants and these inputs, but always sitting that, beneath a framework or a broader overarching thinking around soil management, your soil health principles. This is the broad, I fully support, these are the types of broad strategies that we should be using. And underneath this, yes, we're going to dive deep into specific things and microbes and specific plants and all the stuff we're going to go dig into now. But it sits under this broader thinking. If you can just be following the broad soil health principles at a macro level, you're moving in the right direction for sure. So I put the context of what we're going to talk about underneath this, or things like conservation agriculture. They boil theirs down to 3 principles. Very similar, again, minimizing disturbance, keeping the soil covered with litter, or living roots, and again, plant diversity. So yeah, sitting all of these specifics under that framework. Great time for a question.
We've got a question from on the app. Could you explain how disturbance impacts the presence of AMF?
Yes, sure, of course. So as we can see visually, the mycorrhizal fungi, they grow in these strands called hyphae, these threads. And obviously, if we cultivate the soil, if we disturb the soil, we are smashing, we are breaking apart those fungal networks, those fungal pathways. So physical disturbance cultivation is definitely a #1 enemy of fungus. They really do not like being disturbed and broken up. It will break apart those very tender neural pathways there, the fungal hyphal pathways. Now, of course, this is a nice visual image. Okay, this is on a pine tree. It's not maybe quite so directly relatable to a annual cropping system where we've got a much deeper, clearly a deeper effect here than what we might see in a annual cropping system. But of course, some shallow cultivation or shallow disturbance at the surface, okay, you've still got mycorrhiza below, they can still recolonize. I would argue that even if you did still do some shallow physical disturbance at the surface, as long as you replace that very quickly with a living root and you've got a living plant and some exudates, the fungi will still be there below. Those roots will emerge. They will recolonize. They can carry on the process there. So it's not to say that all is lost, but of course, we want to minimize disturbance as much as possible, but we can also work to try and minimize that negative and replace it with living roots to help rebuild and restore. Okay, so let's dive into plants. Using plants to just get those nutrients out of the soil. Now, this applies to your cash crop, using those plants, but also using companion plants, using cover crops. It all applies. So we have kind of two things here. The plants, as I touched on a bit already, they can release organic acids. These acids can directly solubilize nutrients from the soil. These are acidic materials or hydrogen, those exchangeable things we talked about just before. So this is a direct solubilization, acidic materials stripping the nutrients off the soil, releasing those nutrients directly for plant uptake. And again, if we look in a lot of classic kind of soil science thinking and textbooks, that this was kind of the how we always talked about plants getting nutrients from soils. It was about releasing hydrogen, about releasing organic acids, direct solubilization. Not so much acknowledgement of our friend up here in red, the biological contribution to also helping with this. And now, thankfully, this has changed. There's so much traction and interest in soil biology and the biological processes and understanding and making this work for us. So, okay, it turns out that's half the story. The other half is that plants also release a bunch of different other root exudates, sugars, carbohydrates, very specific signaling molecules, biochemicals that kind of communicate and trigger microbial responses. So we are now feeding and activating all of the living component, those microbes, and let them do the solubilization. And indirectly, they will then release those nutrients for plant uptake. And the key point is to say that microbes, as I'm sure you're all aware, our bacteria, particularly bacteria and fungi, they exist in soils in such diversity. There is thousands of different species of microbes in soil. They are a vast world of diversity. And with that diversity of organisms comes a diversity of tools in which they can use to access and unlock those nutrients. So with all of their various all of the genes that they have inside embedded within them, those genes code for these very specific enzymes that can unlock FOS or unlock zinc or do this or that, et cetera, et cetera. So point being, microbes have way more tools in the toolbox than plants do. There's a whole lot more diversity, a whole lot more genes that code for these things than what the plant has itself. So it's much better for the plant actually to foster them, get nutrients through biology, through their many and varied tools in which they have versus a direct solubilization. I mean, both are important, but the other half is particularly important. So, and again, different plant species have different root exudates. Here we're just looking at the pH as one small example. We have faba bean on the left here. You can see it has very acidic root exudates, making the zone very acidic. That's great for solubilizing certain nutrients. Again, FOS particularly would be a good example. Here we have soybean in the middle, and you can see it's a bit mixed. Different parts of the roots do release different types of exudates. Some root exudates come more at the tips and some a bit further back. So we get this kind of mixture there, you can see. But over on the right-hand side, we have maize, and we can see that the maize root exudates are more in the kind of neutral zone, different plant species. Just really nice visual example highlighting the different species release very different sets, different fingerprints of exudates that are unique to that plant family, even unique to that variety. These can be that specific. Sometimes specific varieties can release very unique exudates versus another variety. So they are highly specific to different plants. So they have different skills, different specialities, different modes of action in which they can access those nutrients. And of course, well, what happens if we bring them together? What if we use this idea of diversity? What if we bring that bean together with the maize and then we get this root intermingling? They're growing into each other's rhizosphere. Well, great, because you know what, the certain nutrients become highly available under acidic conditions. FOS, trace minerals, very available under acidic conditions. So if that maize root is growing into the rhizosphere of the bean, it can get some of those trace minerals that are highly available under acidic conditions. And equally, that bean, it's kind of coming over into this more neutral corn rhizosphere. What nutrients are a bit more available under more neutral conditions? Calcium, magnesium, molybdenum would be another big one. Molybdenum really important for nitrogen fixation for the legume. So it can get molybdenum even more effectively from the rhizosphere of the maize. So when we then start to have this conversation about diversity, we're opening up this opportunity for synergy, where 1 + 1 may equal 3. You know, here's the fingerprint, the composition of the root exudates from the cereal, and those root exudates can trigger and activate and communicate to a certain group of microbes. But as soon as we put another plant in there, well, that plant can speak to different microbes in a unique way that the cereal can't. And then when they begin to overlap and intermingle, there's the synergy effect there. The cereal can benefit from the activation that the legume induced. The legume can benefit from activation that the cereal can only induce. So there's an opportunity there to bring companions together, find these specific synergies. Okay, it's not always synergistic. They can compete with each other, of course. They can be antagonistic and compete, but they can also be very collaborative, and it's about identifying those. So again, great opportunities with things like companion and intercropping, etc. And of course, then going even beyond that into multi-species, multi-species, diversity, of course, much easier in a pasture and a grassland situation, a bit harder to do in a cropping scenario, which is why I think intercropping is great to see that happening. That's good in a cropping situation, but okay, in a cover crop or a grassland situation, of course, we have great diversity. Again, different species, not just different root exudates, which is really an important part of the discussion, but even different root systems, different root architectures, different root depths. You know, all of these, look at the mining of minerals that will be happening down here at depth because of these deep rooters, moisture, bringing that up and releasing that moisture and releasing those nutrients, cycling those nutrients up to the surface areas. There's a lot of benefits that potentially then can arise from greater plant species diversity. And that's a big topic in itself, also not for today. But just to point out that, yes, there's some great opportunities there to, again, it's just really about using more tools. You know, more plant species means more tools that you can use to access, because, you know, this plant is going to be specialized at accessing something versus this plant's going to be better at something else, and this plant's going to be better at something else. And when they all come together, they're expressing that speciality and then collaborating amongst each other. So, okay, well, do cover crops benefit soil biology? Do cover crops benefit the soil microbiome? I mentioned this earlier, you know, roots and root exudates particularly, these are the preferred food source for microorganisms. They love root exudates. They're just so easy to feed on and grow around with the plant, etc. So plants are a great way, their roots and their exudates specifically are a great way to feed the biology, to activate the microorganisms in the soil. And here we have a nice, again, another meta-analysis, a study that's reviewing lots of other studies and pooling that into kind of broader conclusions. And so here we're looking at, do cover crops benefit soil biology? Is cover crops a good strategy to activate and improve soil biology? The answer is yes. We conduct this meta-analysis combining the results of 60 relevant studies, looking at cover cropping effects on soil microbial properties. Overall, cover cropping significantly increased the parameters of soil microbial abundance by 27%, soil microbial activity by 22%, and microbial diversity by just a little bit there, 2.5%. So yes, plants, cover crops, they do benefit soil biology. It's a great way to simply feed. And that's again why the living roots soil health principle is just such an obvious good one. However, you can apply and integrate a living root for as maximum time as possible, the better, for sure. Okay, so here's a really interesting study. This was, again, a hydroponic study, a lab study, but really interesting, looking at this kind of nuance of plant diversity and a diversity of root exudate cocktails. So what they were doing here is feeding a mixture of artificial root exudate cocktails and looking at a microbial response. So we're trying to measure microbial biomass here. And what we've done is we have a control and then we have low and high. Now, low and high means a low diversity of root exudates. So a mixture of 7 different root exudates, and a high, they gave 18 root exudates. Okay, so now the amount of carbon, the amount of energy, the raw amount of energy, the raw amount of carbon was the same in both treatments. It's just that the low treatment was a mixture of 7 exudates. The high treatment was a mixture of 18 exudates. So more diversity of exudates, which of course we would get from greater diversity of plant species. So let's have a look. Let's just start here in the first part of this. Okay, as soon as we add some exudate, some food source, yes, we get a very good response in microbial biomass. Yes, they grow, but this is the low treatment. So that's great. We responded when we gave the microbes, again, the same amount of carbon, but just a more diverse diet. You can see that that induced an additional increase in microbial biomass. So the microbes were responding to a more diverse diet is the take-home message. Now that's happening when we just had one plant species in the mix. Now we repeat this and now we put two plants together and you can see the same effect happens. Here's our control. As soon as we start from, there's two plants now in this mix, we just simply feed a low or high diversity of exudates, artificial exudates. We induce, again, greater benefits from the high diversity exudates. As we move up to then 3 plant species in this trial, you can see here very clearly that that benefit of the more diverse artificial exudates has begun to narrow, has closed, because we now have more plants actually there in the study, and so we're getting more diversity of exudates coming from the plants. So that I'm pointing out that the effect of the plant diversity is now beginning to override the benefit that we were seeing early on with the more diverse cocktail. So just again, really just pointing out, highlighting that yes, that plant species diversity seems to be very good for microbes, and it's because of those diverse exudates, which is their preferred and favored food source. So how can we optimize root exudates? This is a big topic and another big interest of mine. We could also do a whole hour's lecture on just on root exudates if you want, but another time. So let's just kind of have a look at some of the key factors that are that kind of influence the root exudate production. They're both the quality and the quantity. As we kind of just touched on, plant species, like this is definitely the big one. The more different species diversity, the better diversity of exudates you're going to have. So that's a big one. The plant age or the plant growing stage. So we know that plants will release more exudates in the, in the vegetative stages. As soon as the plant switches to reproductive mode, it's putting more energy into grain fill, into flowering, into grain fill, et cetera, et cetera. So there's less exudates in the reproductive stages. Your maximum exudation happens during vegetative growth stages. And again, so that's why grazing is also good, because by bringing the animal in and grazing the plant, where as it's kind of moving through its maturity into the reproductive stages, we graze that and it sends it back into a vegetative stage where it then keeps those exudates turned on. So this is part of the reason we see, good soil health responses from perennials, from grass, animal-plant interactions. Okay, plant nutrition, photosynthesis. So if the plant has a mineral deficiency, if it's limiting in any nutrient, it can't photosynthesize properly. It can't metabolize properly. So that's going to have a negative impact on its ability to grow roots or to produce those root exudates. So optimizing plant nutrition. So again, using your SAP analysis or your plant tissue analysis, making sure you are addressing any limitations, any of the micro minerals, any of the macros, you name it. If anything is limiting photosynthesis, growth, development, and root exudation is not going to be optimized. Neighboring plants, this is a super interesting one. A few studies show that plants really do communicate through smells, through volatile organic compounds. So smells, what we would call smells or scents, that they off-gas these gases, they off-gas these compounds, these volatile organic compounds. And those volatile organic compounds, indeed, they communicate to each other through those. Those can be off-gassed and then land on another plant. The plant detects that signal. It can also change its root exudate profile, thinking that there's a neighbor by who could be a competitor, or it could be also synergistic. These things can also work to their advantage. This is a whole field of research that we need to do with intercropping, understanding how certain companions work better than others through some of these volatile, gaseous kind of communication as well. Very interesting space. Interacting organisms, okay, so of course they can also play a role. Microbes around the roots can also influence this. Pests or disease or insect herbivores or grazing herbivores. I mean, all these things can then change how the plant is stressed or behaving and influences these root exudation composition. And then there's just all of the other kind of environmental type factors that also play a role as well. So things like, light, of course, that's going to impact photosynthesis, temperature, soil pH, soil moisture, soil nutrient content. All those kinds of environmental things are also going to indeed play a role. Okay, so I share this one also just linking quickly back to our discussion about soil organic matter. Do cover crops improve soil organic matter? Are they good for this? Are they a strategy that we could use? And again, we have a good meta-analysis kind of reviewing this and just summarize key points. Cover crops, according to this review, across the board, again, there's a whole host of variation amongst this, but we're just trying to find the broader trends. Yes, they can increase soil organic carbon by around about 15%. Clay soils are more responsive than sandy soils, okay? And again, that's because clay soils have more of those mineral surfaces to form that more stable mineral-associated organic matter, so that's why. Definitely more potential in temperate soils versus tropical. It's harder to build soil organic matter in tropical soils where it's warmer, more cycling of that organic matter. In their study, they found that mixtures did outperform monocultures, better opportunity with diverse systems, and indeed the surface soils being particularly much more responsive as well. Okay, so we talked about this briefly already. I wanted to share this. It's not just also about scavenging the nutrients from your soil, although that's a big part of our discussion today. It's also about just bringing in some free nutrients from the air. Thousands and thousands of tons of nitrogen gas is hovering above every single hectare. There's oodles of it there. You just got to unlock it and access it from the air. And of course, the nitrogen fixing bacteria have the special keys that let them do that. They produce a very special enzyme called nitrogenase that takes that nitrogen gas and breaks it apart and gives it to the plant in the form of ammonia. So yes, we can get some free stuff also from the air. And as we touched on, our legume friends are key players, and they can help and transfer that to our cereal. And it's not just that's one way. Here's a really interesting study that showed that actually with maize and faba bean, they actually showed that the maize itself releases specific root exudates from the maize that actually enhance or activate the rhizobia. So the maize itself is activating rhizobia, even though maize doesn't form nodules and doesn't associate with them, but it does produce exudates that can activate them, as well as other exudates that ultimately support the nodulation process and support the symbiosis there between the rhizobium and the bean. So here we have an example where the maize is specifically helping the bean. They're not competing with each other. The maize is helping the bean fix more nitrogen. Why would it do that? Aren't they competing? Isn't this a threat to me? Aren't you going to be stealing moisture and stealing nutrients from the maize? Why is it helping? Well, of course, there is that end transfer back from the bean, ultimately back to the maize as well. So it's in its own interest to help the bean fix more nitrogen, because then it gets some of the rewards from that. So that's a really interesting one here. Again, kind of similar example, diving a little bit deeper into this. So here we have an example from wheat and faba bean intercropping system. So here we're looking at, for starters, the effects of either a monoculture or an intercrop, so 1 versus 2. And we're looking at mycorrhizal colonization. So for starters, you can see the yellow is just a monoculture wheat, pure wheat. And here we have the mycorrhizal colonization of those wheat roots. When it's just a monoculture wheat, we can see mycorrhizal association is whatever this is, 25 odd percent. However, by simply then measuring mycorrhizal colonization in the wheat again, in an intercropping system, simply having a bean also there present, we see that the colonization of the wheat plant, simply by having a bean companion nearby, actually increases. So we get more mycorrhizal colonization of the wheat simply by having a bean in the vicinity, in the mix. And also, the same is true of the bean itself. If we measure a faba bean monoculture, here's its mycorrhizal association, 40-odd% of the root system. Whereas if we have a wheat companion for the bean, well, you can see that the bean's mycorrhizal association also goes up simply by having a wheat companion. So there's definitely a synergy there of having the two plant species optimizing mycorrhizal colonization. Now, if we then also look at nitrogen fixation, we're looking at nitrogen derived from the atmosphere. Now, this is also quite interesting. This is with and without mycorrhizal fungi. And the study is looking at this for this reason that we touched on earlier, that yes, there can be transfer of nitrogen directly through mycorrhizal fungi into the non-legume. It's for that reason that they're looking at this. So here we have with and without mycorrhiza, gray is without, green with. And now again, under a monocultural situation, with or without mycorrhiza, wasn't really such an impact. They're basically the same. The amount of nitrogen fixation, the amount of nitrogen derived from the air was the same under this scenario. However, when we then had the mixture, the two plant species, and again, with and without mycorrhizal fungi, you can see that now by adding mycorrhizal fungi in an intercropping system, in the diverse mixture, that actually induced greater amounts of nitrogen fixation than without that mycorrhizal fungi. So I'm saying that simply by having the mycorrhizal fungi present, that meant that more nitrogen was getting transferred, and that was therefore becoming a drain. That's becoming a sink. So that had to force the legume into keep on fixing. It would fix more nitrogen than if the cereal wasn't there, because the cereal kept taking that nitrogen from it through the mycorrhizal fungi. So therefore, we are inducing the legume to actually fix more nitrogen under diverse systems than we would under a monocultural system there. Okay, so that's really interesting and a good example of why then in intercropping systems, we should also be thinking about encouraging biology and supporting these biological processes. There are lots of benefits to that. Good, Tony.
Another question from the app. Is there specific information in regards to which species of grasses cooperate best with alfalfa? Is there a difference between varieties of alfalfa?
Yeah, great question. And I'm sure that there would be some grasses that work better than others. I think Timothy is generally a good one. I don't know the answer to this question. But yes, I'm sure that some would be better than others. And I would say for sure, and this is also one of the both challenging and exciting things about intercropping, is that for sure it is variety specific. Some varieties are better in intercropping systems than other varieties. It does go down to that level of specificity, and there's lots of annoyingly knowledge gaps in that space, but at least we know enough that it is a factor, and therefore, okay, we've got to drive that research agenda and try and answer those questions to help you optimize which varieties. So yes, we do know that certain varieties of any plant species, some are better than others within more diverse systems or not. Yeah, for sure. But there's a ton of knowledge gaps on that. Okay, so here's an interesting one. This is, okay, a grassland. This is from Ireland. I know it's a very different climatic condition, but it's just a really interesting example to kind of share. Here we're looking at a grassland kind of production system. And along the bottom here, we're looking at different nitrogen application rates. And we're looking at a monoculture, this is perennial ryegrass, versus some more diverse systems. Now, the mix one is 4 species. It was 2 grasses and 2 legumes. Timothy, perennial ryegrass, red clover, white clover in mix 1. And then mix 2 was again Timothy, perennial ryegrass, just the 1 legume, red clover, but also ribwort, plantain, and chicory. So some other herbs kind of coming into a more diverse. So we've got a 4-way mix and then a 7-way mix across lots of different nitrogen fertilization rates. Okay, so let's break this down. Here's our monoculture, perennial ryegrass. At 0 nitrogen input, at 0 application, here's our baseline. That's how much biomass we could produce with 0 fertilization under a monocultural situation. And of course, if we look at 0 and 0 over here, just simply having those legumes in the mix, we can see that clearly we're getting a lot more biomass production under those more mixed situations. So then we go up to 120 kgs of nitrogen under the monocultural ryegrass, and here's our biomass production now. We've got a jump from that fertilizer application. If we then kind of follow this line across, we can see that, wow, even still at 0 kgs of nitrogen in this more diverse, it's still out yielding the biomass than at 120 kgs. And again, in our mix of 7 species, again, it is out yielding. So let's now go to 240 kgs of nitrogen. Here's our biomass yield under the monocultural situation. Okay, and now we've kind of reached this point here. You can see that we've just slightly under yielded, not too far behind, but we've just under yielded here. Still 0 kgs of nitrogen versus 240, 0. We're only just a small minor yield below. But okay, look over here. In our more diverse mix, we're still on par. So even at 0 kgs of nitrogen with the 7-way mix, we are on par with a 240 kg application of nitrogen per hectare under a monocultural situation. Okay, so again, really nice visual example illustrating the potential there for nutrient optimization, nutrient use efficiencies, reducing our input dependency. And again, there are many more examples beyond just nitrogen. It's not just about nitrogen and legumes bringing in nitrogen. There's all sorts of other synergies and root-to-root interactions whereby in which plants help and support each other. Big one is phosphorus. Again, legumes are actually also, again, because of those very acidic root exudates that we saw the picture of earlier, legumes are very good at, through that acidity, accessing solubilizing nutrients, particularly phos. They also release very specific phosphorus releasing enzymes that can liberate phosphorus from organic reserves, liberate that and make that available. And when the legume is liberating and unlocking and cycling all of this phosphorus, well, indeed, the non-legume companion, whatever it might be, okay, a grass in this example, can then also benefit from that. So again, root-to-root intermingling, the legume is really good at solubilizing phos. If we have a grass or a cereal root system in there with it, it's going to be scavenging some of that phosphorus as it gets liberated, as it becomes available. Okay, so it's not just about nitrogen. We also see other examples, and it goes both ways also with some trace elements. I'm going to zoom in here. This is the same thing as that last slide, but just a better picture. It turns out that grasses, anything in the grass kind of family, cereals, they release a very specialized root exudate these are called phytosiderophores, just a very specialized root exudate that is very good at grabbing onto iron and zinc. So it forms like a kind of lock and key. It grabs onto iron and zinc and pulls it off the soil particles, releases it, makes it available, solubilizes. And so it releases this specialist kind of chelating agent, this phytosiderophores, grabs onto those nutrients where it can then take them up. So it does that for itself. But it turns out that when we have also a legume companion, for example, that the legumes also have a little specialized little enzyme that they can release that also can grab onto, as that phytosiderophore comes out, it grabs onto the iron, it forms this iron phytosiderophore chelate complex. Well, this specialized enzyme then can also break that apart, reduce that iron, and let that iron be taken up. So I'm just sharing another example here where actually the legume is the one benefiting from the exudates of the cereal or of the grass through these specialized phytosiderophores. And here's the visual at the top. Monoculture, okay, this is peanuts and maize, but still it's true. It's good examples of many legumes and cereal type combinations. So we have a monoculture peanut that's clearly iron or zinc deficient. We can see that chlorosis there. Simply by having the maize there as the companion, it's activating the trace elements in the soil through this pathway. The legume is then benefiting from accessing those. These are in the same soil, it's the same study, yet I can't see chlorosis in these peanuts here, for example. Okay, so it's way beyond just nitrogen. There's all these really specific things and specific nutrient interactions that benefit from more diverse systems. Okay. How long should we spend on this slide? Don't worry, I'm going to make it a lot easier. Okay, here we're looking at a bunch of different cover crops, monocultural cover crops, some various combinations, and then an 8-way species mix of them all. And we're looking at all these different cover crops and combinations and how they impact the microorganisms in the soil. Now, we're going to zoom in here. There's all our individuals, and we're going to just take an example and kind of mainly zoom in at the 8-way mix here. So what I'm going to do is we're looking at how these different plants benefit these various different bacteria groups, non-mycorrhizal fungi, mycorrhizal fungi, and protozoa. So I'm going to circle all of the treatments that were the highest. So for example, this one was the highest at stimulating this bacteria. This one was the highest at stimulating that bacteria. This one was highest at stimulating the actinobacteria, et cetera, et cetera. So you can see here that, okay, there are some instances where the monoculture indeed was the best option. It activated the actinobacteria the most. We can see oats, I think it is, were really good at stimulating fungi. Okay, so a monoculture of oats did do the best. But we can see that there's also some good results down here under the more diverse mixtures. We can see here more diversity seemed to be the winner, and certainly in terms of protozoa, more diversity 8-way mix was the winner. Now this is after 2 months. This is at a 2-month mark, measuring the microbial benefits or changes from a 2-month stage at the cover crop. And then now here we have at the 8-month, or is it 9-month? 9 months, sorry. So now we have it at the 9-month. And again, here's our individuals and here's our 8-way mix. And you can see there's this bit of a trend. There's a lot more circles down the bottom here where, okay, over a little bit more time, the more diverse mix was not always, but was a lot of the winner, seemed to bring the most activation or the most benefit to the soil biology. Okay, we can see a couple of examples. So it's not to say that the more diverse systems are always better. You know, there can be good reasons to use monocultures, very kind of targeted and specific applications. I'm definitely not opposed to that. I think that's perfectly fine. But there seems to be a range of kind of benefits for under more diverse systems as well. So yeah, I think really that means use diversity and get down and look at those root systems, see what's going on. I think it's about different species, different roots, and then those different root exudates that really help to kind of drive a lot of those processes and activation of the microbes that we were talking about. So I think it's really important to assess your cover crops and look at the root systems. That's really where it's all happening. It's the roots that are really driving this. It's okay, of course, roots and shoots are linked. They're one and the same, but it's the roots that are really a key driver. So what are some of the things that we can do to improve roots, to manage roots? Okay, within your cash crops, again, this is an area where I think we'll get some really exciting research and better fine tuning of our production systems going forward. It's, you know, again, part of the question we had also earlier, choosing varieties or choosing breeding varieties or choosing species that have lots of roots, high root to shoot ratios. And we could do that for sure cover crops or etc. But we could also do it for cash crops. Choosing big rooting cash crops is going to be a great strategy because you're going to be building soil health, but still having a harvest as well. So however you can choose species or choose varieties that have more root systems, more root biomass, is going to be good for feeding soil biology, for improving soil organic matter, as we talked about already as well. Okay, so that could be, again, perennials, having a perennial phase in the rotation, or using perennials, having perennial systems, again, is always a good strategy. Not something we're going to talk about today, but again, there's a selection of kind of inputs we could use to enhance rooting, and that's like nutrients, phos, really important for root development, sulfur, boron, they're all really important for root biomass production. Could be some biostimulants, humic acids. We talked about that protein hydrolysates earlier with the maize and how that induced a lot of good rooting biomass. So there are some inputs that we could use to kind of stimulate more root production. Maintain a living root. I mean, soil health principle, just keep them there, avoid bare fallows, keep living roots, keep root exudates turned on, minimizing disturbances. We had that question also. Polycultures, we've been through some of that research. It seems that diversity can, in many instances, be very beneficial to support these kind of processes. Same deal with your cover crops. Choose ones that have bigger rooting, bigger root systems, higher root to shoot ratios. Better quality litters, we talked about that, you know, bringing in a lot of legumes into the cover crops or into the grasses. They do break down easy, they have good quality litter, supporting microbial biomass production, supporting that necromass formation, and of course things like livestock integration wherever possible. Grazing techniques, you know, keeping those roots active and giving them good recovery time between grazing, also very important strategies. Okay, so that's plants. Now that's probably the bulk of the section. These next two are going to be a little quicker. Wasn't going to dive as much into these two. Should be good for timing. Okay, so let's look at biostimulants. Here's simply our opportunity just to feed the organisms, to give them food sources, activate them. I put plants first intentionally. I think plants are our number one. We should be using plants and living roots to feed soil biology. I think that is the number one strategy before we move on to inputs and other things, et cetera, et cetera. I think that's the foundation. And then can we fine tune and enhance through inputs and these other things? Sure, yes, we can. I'm all for an integrated approach. Let's use all the tools we can. So, but I do think plants should be the foundation. So what are biostimulants? Often biostimulants are defined as compounds that can stimulate either plant growth. It's usually in relation to plant growth, but they can also be used to induce benefits to microbial growth as well. So compounds that can induce microbial growth or plant growth through non-nutritional means, not really through supplying nutrients, or non-kind of pesticidal or plant protective type means. It's other means in which they are stimulating and activating either plant growth or microbial growth. So what are some of the main categories? Okay, things like humic substances, your humic and fulvic acids, which I'm sure many of you have come across. Amino acids, protein hydrolysates, we talked about these a bit earlier. They can be animal or plant-based. I'm going to say a few more things on that again now. Macro and micro algae, so seaweed extracts, seaweed are your macro algae. All sorts of other plant extracts. Of course, plant extracts are hugely diverse. It could be root extracts, shoot extracts, fruit extracts, water, alcohol type extracts, and there's thousands of different plant type extracts that could be biostimulants. Microbial inoculants themselves, so biofertilizers, are often considered under this kind of category, or a bunch of other even inorganic substances or nutrients that are not considered essential for plant growth. So things like silicon, selenium, cobalt from some plants, sodium for some plants that don't have an essential requirement for those, but they can be very beneficial. Now, the difficult thing with biostimulants is that we're talking about classes of compounds that are extremely diverse. There are hundreds, if not thousands, of different compounds that can have biostimulant effects. And of course, therefore, all of those different compounds have so many different modes of action, multiple modes of action. So they're very complex to study. It's not just one thing and it's one left or right, one or two. It's not simple. It's thousands of compounds, thousands of mode of actions, thousands of interactions. So therefore, they're a very complex thing to study, and that very kind of rapidly muddies the water. But there's some exciting things, again, in the world of science happening. We typically have thought about biostimulants and their effects on the plant phenotype. So here I'm talking about things that you're kind of actually interested in. Or do they improve root or shoot growth? Do they improve whatever yield, flowering? Does it improve photosynthesis? Does it improve stress? These kinds of things is how we've typically and historically studied biostimulants, and that's all great. It's what we all want to know, practically speaking. But we're going through this transition where we can also then study the effects of biostimulants at a cellular level, or even more specifically at a molecular level. So here I'm talking about at a genetic level, what do the biostimulants do and which genes do they turn on or off, getting deeper and deeper into the way in which we study these things. And this is going to be very useful because the more we understand that level of specificity, that specific gene expression, for example, it will help us overcome some of the variability or the problems with studying biostimulants. When we really have that deeper understanding at the molecular level, we'll be able to translate it better in terms of field application or field applicability, et cetera, et cetera. In the context of benefits of biostimulants, I'll read you this list, and it kind of sounds like all of these claims and things you'll hear on all sorts of product labels, on every product label, whatever: reduced nutrient leaching, seed germination, nutrient uptakes, crop establishment, stress resistance, improving shelf life, improving roots, crop performance, stimulating immune systems, improving defense chemicals, heavy metals, you name it, all these kinds of claims that we might see on products for these types of things. Just to let you know also that, this is a review study of biostimulants, that to say that in a lot of those kind of claims, yes, there is actually good evidence that they can do these things. Biostimulants can, there is studies to support that they can. It's just, again, these questions of how consistently do they do it? That translation into field conditions is not always so perfect. But just to say that there is a foundation to this, but okay, we've got to fine tune that a little bit more in terms of improving that applicability, etc. And just then bring it back to a nutrient context. Here's an example of seaweed. Just quickly to share this, it's a really interesting example. Like, what is seaweed? When we talk about seaweed extracts, what are they made up of? Well, it's mostly carbohydrates, protein, and minerals, the essential nutrients, the macro or micronutrients. You can see this is a very, very minor part of what a seaweed extract is. It's not really about the direct fertility. It's not about the nutrition that it's supplying. That's a very minor part of what a seaweed extract is. It's all of these other kinds of compounds. However, if we look at some of the studies that have then assessed effects of seaweed extracts on a range of different plants here, maize, canola, tomatoes, canola, wheat, canola, soybean. If we have a look over here, what were some of the findings? We saw enhanced NP and K uptake. We saw enhanced P and K concentration. We saw enhanced fruit calcium. We saw increased manganese, copper, and zinc in the root and leaf. We see increased manganese, copper, and magnesium in the whole plant, enriched grain potassium, stimulation of root and shoot nitrogen and sulfur, enhanced nitrogen, phos, potassium, sulfur in the grain, enhanced nitrogen, phosphorus in the straw. Look at all these benefits to the nutrient content of the crops, despite the fact that you're delivering an insignificant amount of nutrients to the plant. It's not a direct nutritional benefit. It is more of these indirect kind of biostimulatory effects, which we are untangling more and more, that are actually bringing the benefit to that. So just to point out, there's a lot of work looking at the role of biostimulants to help nutrient use efficiencies. This is one of the areas where they can help. There's good evidence. There's all sorts of claims again, but where there's pretty good evidence is that biostimulants do help with improving nutrient use efficiencies, and they do help with a lot of the stress resistance, particularly drought and some of the temperature stresses and these kinds of things. So some of those environmental stresses that there is a bit better evidence on. This was that slide, Rob, that I thought I had in the other presentation on some of the protein hydrolysates. I was just going to share, some of these examples of materials that can be used. Again, fish wastes or whatnot, blood proteins, whey, poultry, hatchery waste, vegetable proteins, soybean, alfalfa, other agro wastes, stubbles, husks, pomace, et cetera, et cetera. And I share this, this is then just saying, we can make protein hydrolysates out of these waste materials. We can digest them, find specific enzymes, digest them, break them all down and form these nice biostimulant type things that have good growth responses. I wanted to share this again because again, in many circles, there's a great sense of kind of skepticism about biostimulants. I see a lot of criticism of them, or just another wave of products that farmers have to buy and be addicted to, and et cetera, et cetera. There's this kind of big discussion about some of the negatives or the critique of that. A very important conversation, absolutely, for sure. One that we must have, but I just think this is also a really interesting example, the protein hydrolysate specifically. I think this is a really good example of biostimulants. Why shouldn't we take waste products? They're waste products. Let's turn them into something useful. This is good for the environment. We don't have to put this stuff in whatever, landfill or in the ocean or whatever. So I think there's a lot of really interesting applications of taking waste products, breaking them down, digesting them, identifying unique compounds that could help with improving nutrient use efficiencies. So I think that's a good example. It's not always these biostimulants being the latest snake oil or whatever. I think there's some very good applications and some good thinking about how we could maybe use those. And we saw the slide earlier, again, for the obvious reason that we get this whole metabolic shortcutting thing, and we can break them all down, but also these things, these more complex molecules are very good for plant efficiencies, as we talked about. And again, this idea of supplying nutrients that are embedded with carbon-based inputs, organic sources of nitrogen. This is amino acids, nitrogen with carbon, these kinds of things, these kinds of carbon-based nutrient complexes. This is a great way to stabilize that nutrient. If that was nitrogen, it's not going to be so leachable when it's bound up within carbon. It's not going to volatilize off so easily as ammonia gas when it's bound up within carbon. I guess carbon-based fertilizing strategies are a really good way. So again, this idea of combining humics and things with your fertilizer granules, I think is a really good idea. Okay, sure, great timing.
Okay, we got a couple questions for you now. You mentioned nitrogen use efficiency to be 40 to 60%. Can you expand on this basis for this estimate? Is this a regional assumption, example within Canada or North America? And is it under certain cropping conditions?
Sure. Okay, there's a lot of work. There's a lot of studies that would support that number, that figure from a whole range of different soil types and climatic conditions. And I did bring the caveat there that it is an average. Definitely in some situations you can see much better, higher nutrient use efficiencies, and in some situations also lower. But across that kind of variation, this figure of 50%, yeah, it's a bit of a middle ground. It's an average figure, but it's definitely in that vicinity. And yeah, that would be relevant in here and many countries around the world for sure. Yeah, there's a lot of evidence to support that figure. Yeah.
Thank you. And the next question is, are biostimulants better applied by foliar application or in furrow?
Ooh, great question. Most of if you really dive into a lot of these studies on biostimulants, most of them are using them through foliar pathways. So that seems to be, yes, where they do have more benefits for the plant. Because of course you're applying it directly to the plant. You put that biostimulant in the soil. Now you've got all of those microbes who are going to grab it first. So you will, if your goal is to apply benefit for the plant, yes, it's a foliar pathway. Now, if you're trying to use biostimulants to feed microorganisms, however, to feed the soil biology, the soil biostimulants, well, then in that situation, it would be better in the furrow. But generally, most of that kind of work is looking at foliars. But yeah, they go well in the furrow too. Like liquid inject with some humics and molasses and fish and these kinds of things injected with that seed, I think is a very, very good idea.
Just one more question, Joel. Do you have any easy or practical ways for producers to test these biostimulants? Because everybody and their chicken has one. So
yeah, that's a good question. And that's a tough one. I feel for you. There are so many products out there and there are just more and more and more and more forever on the market. And of course, it's hard to wade through the sea of that and identify which are the better quality ones. So there's no easy answer to that. Part of the answer is I would encourage and foster peer-to-peer learning. You guys working together and collaborating, take recommendations from other farmers who are using certain things and having good results. That's a great start. Recommendations and sharing what works for you versus not. So I think there's a social peer-to-peer element, but at a more practical level, I would say that germination trials, a quick kind of germination trial seed is probably the quickest and easiest way. So and that could even be a simple little seed treatment and kind of Petri dish type germination trial, or again, get some of your soil in a tray and put those seeds in your soil type as well, and then apply that biostimulant and kind of look for germination and quick establishment. That's going to give you a bit of an idea within a week, 2 week kind of window. I would say something kind of like a quick germination trial. That's giving you a bit of an indication of its biological properties. Is it good for biological systems, biological organisms? A quick germination trial would be the easiest way. Yeah. Yep, is there a question? Yeah, sure. For the listener.
Yeah, just on that biostimulant page, you had kind of the top level where it's the base element and then went through that refining process.
Yeah.
In your opinion, do you think biostimulants are better kind of in their whole form, like a whole grain kind of approach versus a refinement approach when you're using it, say, for a soil application?
Yeah, okay. So that depends a little in the sense that if we were to think about these in their whole kind of context, you do actually want to break them down a little. You want to go through that digestion process to make them get that mixture of compounds, some more bigger and complex, but also some more smaller. And it's that perhaps that more diversity of those compounds that overall kind of helps. So I think you do still want some kind of digestion and breakdown, just not all the way to the absolute smallest and simplest bits, but back at a stage where you've got a mixture of some more complex things and some smaller things, what would be called a partial digestion, in other words. So I think, yeah, you do still want some of that kind of breakdown process to have occurred rather than a full, complete, undigested material.
Joel, you showed like whether it's humics or cropping mix or systems or all these different things where you've increased microbial activity in the soil and all the charts and stuff showing sort of 2 months later. Is there any type of effect like the next year, once that crop is done and left void? Are we getting any type of long-term effects or anything, or just a small blip that results in nothing?
Yeah, good question. Yeah, that's a great question. And it's a mixture of answers to that. Yeah, you're right. In many instances, some of these effects can be very short-lived on soil biology. They're going to be activated at that time, at that point, of course, then they're going to go through the winter and completely go dormant and whatnot anyway, which often, yes, can completely reset the benefit. So yes, sometimes these are then being used more in that short-term way. You're trying to get benefits for that current season. But I would say that that's not always the case. I would say, yes, there are some opportunities where you can see longer lasting effects. And there's some work that's been kind of done on that, for example, with even things like cover crops, where you then might grow that cover crop in 1 year, but you can then still see, some of those other benefits the following years. So I would say there are instances where it can be a bit more longer term, but in many, it is about tools to manage the current crop and the current season. It's a bit of both in that one. Yeah, that's a tricky one. It's very context dependent. The answer to that one.
Hi, Joel. You explained to us the role and benefits of grasses and legumes in a mix. How do you classify the chicory and plantain, and what do you see as their possible benefits in a polycrop?
Yeah, for sure. I mean, so they've got those, deep roots has got to be a big part of that. I think they've got those nice big tap roots. And okay, that can bring benefits to soil physics and the opening of the soil structural aspects, better infiltration, drainage. I mean, you name it, there's some of that element to that. But beyond that, really, it's a different plant families, it's a different species. So it is going to have very unique or very specific root exudates unique to that plant. And okay, we may not know exactly what the specific mechanism is, like some of those ones that I was going through there with the phytosiderophores and blah, 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 all that kind of stuff. We may not know these very specific mechanisms, but it's likely that they have some very unique root exudates that they are producing and causing some of that benefit. Yeah. They're good accumulator, and again, that taproot, good accumulator of subsoil nutrients, bringing them up, also recycling them as well. So. Okay, we're almost done. This section's not too long, so you'll be happy to know. Okay, just after 5, I think that's good. We're going to be a little bit, maybe 10 more minutes or something. Okay, so lastly, biofertilizers. What about this idea of actually putting the organisms into the soil? And this is another one of those hotly debated discussions. So there's 2 schools of thought here. One says, do we just feed the organisms that are already there? Let's just use plants, use those root exudates, use the biostimulants, just carbon compounds, use food, use energy, and just feed the organisms that are already there. And I think that's a great strategy. Another school of thought says, well, maybe there is scope to introduce new populations, apply microorganisms, apply biofertilizers, and introduce new populations of microbes into the soil. I think both strategies are good. Again, I would endorse that our number 1 strategy should be feeding the native ones. Let's just feed the locals who are already there and already adapted. That obviously makes sense. So that should be our primary strategy. But no, I'm not opposed to introducing other inoculants, especially when they're things like compost extracts, for example. Compost that you can make on farm. So you're like these DIY type things where you're culturing those microbes, you're making them in your local micro environment. So clearly they can grow in your environment if you're making the product, that kind of a thing on farm. So I think, again, that would be my preferred use of a biofertilizer, and introducing new populations where you're making them yourself. Something like a compost extract would be a perfect example. But sure, is there scope for bugs in jugs and some other, again, commercial products? Sure, they can also have some benefits. If you're specifically looking for like maximum phos solubilization, well, there's going to be certain very specific microbes who are really good at that, and you can put them into the furrow and they're going to be better at solubilizing phos to get the crop established than maybe something more generalized like a compost extract. Or do you specifically want N fixation? Okay, put an N fixing bacteria in. So again, I think there's scope for some of those products, but again, I would start with feed the locals. Then if you're using biofertilizers, make them yourself, culture local species, and kind of third and finally, using a commercial kind of product. I think that's how I would approach it. I think there's scope for both, but again, our core, our foundation really should be feeding the locals. And again, we have this fierce debate though about introducing new microbes to the soil. So we have, on one side, there's this criticism by putting a couple of microbial products in the soil is a drop in the ocean. You know, like spitting, saying, well, I'm going to spit in the ocean and raise the sea level. You know, the amount that you're introducing into the soil is absolutely insignificant compared to the vast billions of them that exist in that soil profile. So on one side, there's this argument that says it's a waste of time, waste of effort. But I think the nuance that kind of argument is not tapping into is this idea of, well, are we trying to change the whole soil microbiome? Are we trying to change the biology of the entire top foot of soil or whatever? Well, actually, no, we're not trying to change the entirety. Where these microbial biofertilizers can be used is, for example, very specific, very targeted around the seed, around that seed treatment, or again, injected into the furrow, where you're just trying to put microbes in that micro zone around that seed, around that root system. You're not necessarily trying to apply them and dilute them out through the whole soil, through the whole foot of soil and change the soil biome in that sense. It's not really what you're doing with them. Your goal is to be very targeted and put them right there around that seed and let them simply, you only need a small amount. Once that seed germinates and that root emerges and those root exudates begin feeding, that's the job done. Let the plant then do the rest of the work. The root exudates will then feed that inoculum and it will grow with the root system as it grows and emerges and establishes. So it's, I think, wrong and false to make that argument. I think you're not really trying to do that. It's really about being much more targeted around that seed and maybe changing the rhizosphere biome. Yes, you're trying to change that, but you're not necessarily trying to change the entire soil microbiome. So what are the types of biofertilizer we're talking about here? What types of things are there? Okay, again, I mean, compost is a biofertilizer. Manure is a biofertilizer. These things are introducing microbes into the soil. So we've been doing it for a long time. It is a somewhat normal thing for us to do with the use of compost and manures. And we seem to have a lot of criticism and flack at bugs and jugs and this kind of idea on that spectrum, but we do use compost and manures. It's a similar thing. However, yeah, compost and manures are tried and tested and true. And there's a lot of organic matter, there's carbon, there's a vehicle to introduce the organisms. It's not just about applying the organism. The compost, the manure, it's got all that carbon, all that organic matter, all that nutrients. It is a vehicle that helps the biology stay alive, feed, and establish in the soil. So yeah, they're very good, particularly. But okay, can we make things like compost extracts, compost teas, compost slurries? Sure, mix liquid options to use as seed treatments or inject into the furrow. Yes, I think that's a great idea. You've got all of these other anaerobic ferments I see. So things like, indigenous microbes, again, harvesting microbes from your soil type and kind of culturing them up. Again, I think that's a great strategy. But you can also take these to these like fermentative type approaches. So using kind of various lactobacillus type things like yogurt or whatever, like many in our gut, these kind of fermentative types where we can culture some of those organisms. Seems to be some interesting work happening with those. And then, of course, again, I would be starting with all this top list. And again, the commercial products would be at the bottom of my list. Okay, you might want them for a very specific focus, like I said before, N-fixes, P-solubilizers, mycorrhizal fungi, trichoderma, that's a fungus, predatory fungus. It's fungus that eats other fungus, so it's good for disease management. So if you were specifically looking for that specific function, then yeah, maybe you'd want to use something like that. Other endophytes or other consortiums, so mixtures of microbes, not just a single species, but a mixture. It might still be a commercial product, but it's a mixture of some of those different microbes could also be an option.
A question about the commercial products. Do commercial seed treatment products have significant effects on the natural biome?
Yeah, okay. So that's also one of the, again, concerns and criticisms, and it's fair, important debate to have. There's a concern around, okay, two things. One, okay, there's an argument that says, oh, these products are a waste of time. You apply them, they don't outcompete the locals, they don't establish, they're a waste of time. And sometimes that can absolutely be the case, for sure. On the other hand, there's this concern that, well, yeah, don't use these commercial products because you may be introducing alien species, which will then run rampant and take over, and also cause imbalances or problems with that soil biome. So you get two opposite sides there. On one hand, oh, I thought they were ineffective and they don't establish and they don't remain. On the other hand, we're concerned that they're going to take over and become alien species and dominate. And sure, both of those things can be true. It's not has to be one or the other. It's very context dependent. It's very specific. I would say this, if you look at a lot of those commercial products, generally the species that they are using are the more the generalists, the broader organisms that have been identified that are fairly ubiquitous. There are many microbes that exist in the soils in Australia that are the same here in the UK. Like there's a huge percentage that are universal, and then there's a fraction that's also very specific and etc. So if you look at some of the commercialized products, generally they're going to be leaning towards some of those more generalized, common, ubiquitous species. So maybe not quite such a concern, but yes, that is a debate and a discussion to have. Yeah, for sure.
And another question is, how do fungicides affect the strands of hyphae in the soil?
Sure. Well, a fungicide, of course, is going to compromise fungus. And so it can have a negative effect. The answer to that question is every fungicide, every chemical has a specific mode of action. And the answer is very simple. If the organism is sensitive to that mode of action, then yes, it will be compromised. If the organism is not sensitive to that mode of action, then it will not be. So a fungicide, for example, can be very specific against affecting certain fungal species, but there will be others which are not affected if they're not sensitive to that mode of action. So it's a very specific thing. It depends on the chemical and the fungal species in question as to whether or not it's damaging. It's not by default that it is always damaging to the fungus. It really depends on that specific mode of action and this fungal species or the fungal strain even. Yeah. Looks like you have another one.
There's a follow-up on the first question about, it was more about antifungal and anti-worm products.
Antifungal and worm, like is an animal?
Sorry, the first question about the commercial seed treatment products. I was asking about antifungal and anti-worm seed treatment products. Do they have an effect on the natural biome?
I mean, things like, obviously, I'm not sure I totally understand the question, but I mean, things like livestock dewormers and things can then have an effect on the biome in the soil, yes. So that definitely, but I'm not sure I totally understand the question otherwise. Maybe I'll jump in and read that one and see if I can comment on later in the app. Okay, so then there's this big question mark, and I think this one's quite interesting. I mean, is this a natural process? Is this normal? Is this natural to be applying microbes into soils? Does this happen in nature? Many, again, people raising alarm about use of these kinds of products. Well, actually, introducing microbes into the soil is a very natural and normal process, and plants have been doing it for millions of years through their seeds. It turns out that seeds themselves are a vector. We have lots of microorganisms that grow in and on the seed. This is the seed microbiome. A lot of very interesting work, research happening in this space. So it turns out when the plant is growing, yes, there are microbes that live around the roots, as we know, but there's microbes that live on the leaf or in the stems or on the flowers or on the fruits or on the grains. There's all these microbes that live on and in the plant. This is called the plant microbiome or the phytobiome. And so when the plant is maturing and producing grain or fruit or seed, producing the next generation, some of these various microbes will actually travel up the stem through the internal organs of the plant, and they actually get embedded into the seed. And of course, when that seed drifts off into the air, into the water. Those seeds might be carried far with animals stuck on them and all that kind of thing. That seed, when it falls to the ground, it has microbes in it. And those microbes, yes, they are getting introduced into a new landscape. They are aliens into that system. So yes, there is a mechanism already by which this does happen quite naturally. And indeed, they're very important. It turns out these microbes that come from the seed play a critical role in seed germination, in the establishment of that plant. It turns out that they have a very important role to help the plant, help the seed kind of communicate or talk to the local soil microbiome. So if I could use the analogy, the seed microbiome, they start the process, they set the table. Okay, they're the ones that germinate first, they start to grow, the seed microbiome grows first around that seed, and then they kind of open the door, open the dialogue to the soil microbiome, where then the plant and the soil microbiome will carry on that conversation through then germination and onwards. And what do we find through that process is, yeah, the soil microbiome, it pretty much stays in the soil, it lives around the root system, and those species that came from the seed microbiome, they actually do remain and they migrate upwards, where they again colonize up internally of the rest of the plant biome and repeat that cycle. So yes, there is examples where microbes do this naturally, plants do this naturally. And of course, we as humans have been trading seeds and moving seeds around the world for also many, many thousands of years, and taking seed microbiomes into new environments already. Like the horse has bolted from the stable, as they say. So yeah, I think it is a very natural process. Not to say that we should therefore disregard any caution. I acknowledge that maybe the concerns around the introduction of alien species that take over and induce imbalances, really important just conversation to have, for sure. But there is an element of this that also happens quite naturally. So, and that's just a nice image to share of what I was just talking about. You've got the seed microbiome internally. They're really the ones that germinate first. They're the ones that colonize the seed and the emerging root. And then over time, they help the communication with the soil microbiome, who then carries on and colonizes, and they themselves go back up into the plant, as we were talking about. So there's a lot of really interesting work happening about seed microbiomes at the moment. Yep, sure. Sure. Yeah. Okay. So the question for the listeners at home was, how do you know whether to use a biological product or not, or just to use the soil health principles, take the broader approach, encourage soil health, encourage the native organisms? How do you know if you need to use any kind of a product or a biofertilizer? Or the yeah. Yeah, it's a good question. And how do you know? How do we know? Again, like I said, I would always start with the idea of feeding the locals, using plants, using roots and root exudates to just feed who's there. So yes, I would support the idea of the soil health principles, for example. Yes, that should be your foundation. That's where you should always be starting. How do we know? We don't really know if you need a biofertilizer or not. There's so many knowledge gaps. However, many people are using them. Many people seem to be seeing benefits from them. Okay, we know that cultivation hasn't been great for soil, that we've compromised soil biology. We know that some of our fungicides or pesticides can compromise soil biology. We know that certain management practices, growing monocultures, narrows that microbial diversity. We know that many practices have had a negative impact. So there is an argument that says maybe we need to replenish and repopulate. But at the end of the day, I guess it's a little bit of your own experimentation. And if the cover crops and the soil principles are working for you, then great. If you want to play around and try other things, then no harm. I think case by case scenario. Yeah. Okay. And just to also point out, I just want to share. Again, just this really interesting study to say, I've talked a lot about the, root exudates and plants being the driver of the soil biology, plants feeding them, plants exudates. I have this big focus on the role of plants, which is really important. But just to also acknowledge that the microbes themselves in the soil can also change plants. They can also influence that root exudation process. It's very much a two-way street. It's not just that plants are the controllers and the drivers. Microbes are also controlling plants. It turns out that many microbes in the soil have an influence over flowering time. They can induce, those reproductive stages and transitions. So microbes in the soil can change the phenotype, the expression of the plant. Things like flowering time would be one good example that we know very well. So microbes do influence the plant too. It is very much a two-way street. And this was a really cool, interesting study that kind of highlighted this, whereby they were growing the one plant. This is again a hydroponic nutrient kind of solution study. They were growing the one plant in a split root design. So one plant and half the roots into one growth chamber and the other half of the roots into the other growth chamber. And what they did is take some soil and make a soil water extract and put some microbes into just one of the chambers. So of course, and not into the other side. So what do those microbes do? Of course, they all colonized the root system and started receiving all of those root exudates from the plant and started growing around that root system that we've talked about. But it turns out they also are releasing their own microbial signals, their own microbial metabolites, these signaling molecules that the plant is also receiving messages from the microbes. It's not just the other way. And so actually they were sending these signaling molecules up, triggering changes in plant growth, in photosynthesis, in plant physiology, which then induced changes in the root exudation pattern of the plant. So the microbes were able to change the types of root exudates that the plant was producing. And so they could measure that over on this side by harvesting the solution culture and measuring the root exudate changes in this side, even though there was no microbes added here. So it was more of this signaling, this systemic effect, where on the one side they induced changes in the plant development, which we could then measure on the other half of the growth chamber. So just to point out, yeah, microbes, they also play a really big important role too. And again, in summary, I think it's just the easiest way to apply these is a seed treatment. It's cost effective, you only need small amounts, or again, kind of injection into the furrow. Again, it's not about diluting these through the whole soil profile and all that kind of thing. It's just targeted applications onto the seed, maybe injected into the furrow. That's all we're trying to do. Tweak the microbiome around that seed, around that seedling, around the root system as it grows and develops. Okay, so in summary, last slide. Soil nutrients, yes, they are often plentiful. Okay, again, less in a sandy soil, but if you're anywhere else above that, you've got a lot of opportunity to use and leverage the fertility that is already there. Fertilizer inputs are often inefficient. We talked about some of those inefficiencies, 50-odd%. FOS is notorious. FOS will lock up in the soil, can be as low as 10 to 20% phosphorus use deficiencies of what we actually apply gets into the crop. It can be very low. So there are problems with many of our fertilizers. So yes, there is therefore the opportunity to leverage that fertility, use biological processes to access all of those total nutrients that we talked about at the beginning. And again, I talk about these things underneath the system design, the systems approach. Again, the soil health principles, conservation ag, whatever we want to talk about, take that systems approach, use many tools, livestock integration, whatever. It all sits underneath that. But okay, sure, plants, it is those living roots, those root exudates, they are so key to foster microbial interactions, let those microbes unlock all those nutrients. And again, monos or mixtures, I don't mind. Living roots is, I think, more important. Yes, I think there's good arguments for diversity, but sure, monoculture of roots and root exudates can still be beneficial. Feeding biology, stimulating them, it's all about carbon-based inputs and those biostimulants, humics, fulvics, fish hydrolysates we talked about, amino acids, seaweeds, all those kinds of things. They're perfectly good sources. Molasses, just anything that's carbon-based. And then inoculants where relevant. And again, targeting those as seed treatments or liquid inject. And again, leaning towards the preference of DIY inoculants versus commercial products as well. Okay. Questions? 5:30, not too bad.
Okay, I thank you very much, Joel.
Thank you.
And this year, we are presenting each of our speakers with a custom carved charcuterie board. So thank you
Wonderful.
very much.
Awesome. Thank you. Cheers. Thank you very much. Very nice.
We would like to invite everybody here to a reception starting at 6 o'clock. There's no formal agenda. It's just some networking, some sharing ideas. We've all got 2 years of pent up networking that we've got to get out the next few hours. For those joining us tomorrow, registration is at 8 o'clock and the program is starting at 8:30.